And Caret, I don't think has any specific, I mean, like, I think as long as you have more than 3.0 R, it should be fine. Microbiome data, theory and practice. Tatiana and Sabrina, can you hear me okay? Like, is there everything? Can you see my slides? Okay, yes. Great. Perfect. Great. Good to go. Thanks. Yeah, so hi everyone. My name is Sambhawa Priya, and I'm a PhD candidate in the bioinformatics and computational biology program here at the University of Minnesota. And I'm doing my doctoral research in Dr. Ron Bleckman's lab at the University of Minnesota, where I work on looking at host microbiome interactions in human diseases, and I develop machine learning frameworks to understand that association. And so in today's conference, we are going to cover some theoretical aspects of machine learning, as well as we'll do some hands-on tutorial on using machine learning on different microbiome data sets. And yeah, so hopefully, you know, even if you've never done machine learning before, I think we'll cover a broad range, and hopefully, you know, we can all figure this out together. So let's so first introducing the team. So as I just introduced myself, my name is Sambhava, and I'll be conducting session 1 of this tutorial, where in first part of the session I'll talk, you know, introduce the concepts of machine learning, you know, in general, the theory concepts, notations, and so on. And then in part 2, we'll do an R tutorial on you know, applying machine learning on some microbiome data sets. And then then in session 2, Tatiana Lenskaia, who is also a PhD candidate in bioinformatics and computational biology here at University of Minnesota, and she'll be doing introduction to bacteria phage interactions, talking about some, you know, motivation and background on this, and, you know, the theoretical aspects of how we would apply machine learning on these problems. And then she'll do a part 2 hands-on tutorial on using R and Python to look at some of the bacteria phage interactions. And then we have Sabrina Arif here, who just you know, is a first-year PhD student in microbiology program here at the University of Minnesota, and just started in Ron Bleckman's lab, also looking at investigating host microbiome interactions and diseases. So, and she will be our question and answer moderator today for the session. So feel free to post your questions in the Q&A chat, and Sabrina is gonna moderate that, like, and you know, sort of relay that to you know, either me and Tatiana. All right. So let's jump into the part 1 of this session. So yeah, so here, you know, basically we're going to cover an overview of machine learning, terminology, notations, you know, what are types of machine learning algorithms, what are the steps in, you know, to be executed in a machine learning task, and how do we evaluate the performance of a machine learning algorithm. So yeah, so let's look at some of the overview and concept stuff here. So what is machine learning? Let's look at some of the formal definitions here. So it's a computational method based upon statistics for discovering patterns in a data set and making reliable predictions about similar new data. And there are different synonyms that are also used for this, the data mining, computational intelligence, and pattern recognition, and so on. And some of them mean slightly different things, but overall, come under the umbrella of machine learning. And here's a more, formal definition of machine learning. So it's a computer program which is set to learn from experience E. So here we're talking about sort of past data, or like the training data with respect to some task T, where T could be a problem, whether it's a prediction problem or a classification problem, and some performance measure P. So we are going to measure the performance of how this machine learning program is working. So if its performance in the task T as measured by P improves with experience E. So yeah, so that was a definition given by Tom Mitchell in 1997, and I just wanted to sort of give an overview of that. So so in machine learning has been used broadly in biological studies for prediction and discovery. So basically here what we can see is different omic data sets. So we can input one or multiple different types of omic data sets. Here you can see genomic data or transcriptomic data, metagenomic data, metabolomic data, and so on. And their corresponding metadata, right? And sometimes even some prior knowledge that is like domain specific knowledge, such as functional pathways and so on. So all of that that goes into the machine learning algorithm, and to sort of build machine learning models to perform prediction and discovery. So basically this is to show that machine learning can capture like complex biological systems and integrate different data sets to generate predictions. And as I mentioned earlier, you need not have multiple data sets in machine learning model, but that it can incorporate more than one data types. Yeah, and so there are different applications of machine learning in different fields of biology. So for example, in genomics, both human and microbial genomics, there are different ways machine learning has been applied to predict genomic features, especially those that are hard to predict using current approaches such as regulatory regions. So using machine learning to predict the sequence specificities of DNA and RNA binding proteins, enhancers, and regulatory regions, then classifying different cancer types using gene expression data, or predicting the incidence of colorectal cancer using microbiome data. So those are just some examples. And then there are examples in other fields like proteomics, so for example, to predict the structure of protein by analyzing amino acid sequences, or in system biology using genetic networks and signal transduction networks and metabolic pathways, and for example, predicting diagnosis using the stroke diagnosis using classification on neuroimaging data sets. So there are varied types of data sets, different different biological questions, and this just to show that machine learning is versatile in applicability to different types of data and different types of questions in biology. So,
so would you mind if I interrupted for a moment, just before you get too far? In case any of the attendees have to leave early, they're wondering where they can find the recorded this recorded session later on.
Oh, yeah, that's a good question. I don't have the link for that yet, but I believe that if you registered, then Belle Hansen most probably will send out an email from from who you got the email for the workshop registration contents and such. So she will send out an email with the link of the recording, which I believe should be available for a month or at least a few weeks. So.
Okay, thank you.
Cool. Any other questions? I see some. Anything else, or it's okay to go?
There are a couple more questions, but they're specific to installation. So I think maybe Tatiana will be able to cover them in the chat.
I'm commenting, no worries. So yeah, some of you.
Okay, I see someone raised their hand. Maybe just it's. Could you please just write your question in the Q&A, and we'll get to it. Okay, moving forward. So, one thing I want to say. So before we dive into machine learning, basically, I just want to make an important note that it's very important to first understand the biology and the experiments from where the data is coming, and before you start throwing models at it to build to do different predictions or whatever machine learning task. So we want to start with biological questions and then apply machine learning, and from there we can get biological insights, and we see whether it makes sense, and then it's kind of an iterative process. So you apply the machine learning, tweak the model, look at what insights you're getting. Is it biologically relevant, and so on. Okay, so with that note, let's dive into some concepts and terminology of machine learning. Feature. This refers to an input variable that is used in making predictions, and it's also referred to as predictors or covariates. So these terms, if you come across, it's basically interchangeable with features. And prediction or outcome, so it's the machine learning model's output. So it could be categorical. That is, whether if the prediction is about determining whether it's a cancer or a non-cancer sample, or if there are different disease subtypes, or it could also be continuous, such as body mass index or blood pressure, for example. And then there's classification, which is categorical prediction. And there could be binary classification. So when you have to predict, between two classes, and then as is self-explanatory, the term multi-class classification, when you have more than two classes, and you have to, sort of identify samples or predict samples in different, more than two classes. And then regression refers to continuous prediction and involves real values. And we'll see a little bit more of details on classification regression a little bit later. And then model. So the model is the representation of what a machine learning system has learned from the training data. And yeah, there are two terms which sometimes get confusing. So one is hyperparameters and parameters. So hyperparameter are variables that influence the way learning algorithm works, right? So this needs to be identified and set before creating, or rather for creating the machine learning model on the training data. And parameters, on the other hand, are the variables that define the model that is learned by machine learning. So that's those values are set as a result of the machine learning process. And so this is learned from training data, and then it is used for performing prediction on test data. So we'll see some of those examples also as we go further in theory and then in the implementation. Okay, so before we jump in, is everything looking good? And is there any questions so far that is relevant to our theory aspects? Okay, so let's move on. So so let's look at the different types of machine learning algorithms. So there are three main types, and first one is the supervised learning. And in this type, the machine learning algorithm is trained on labeled data. So and the aim of supervised learning algorithm is to try to model relationships between the target prediction output and the input features, such that we can predict the output values for a new data, right? And there are two main types, classification and regression. And we'll cover this a little bit more detail in just a little bit. And then there is supervised learning. So where there's no predefined class labels that are available. So it's like kind of opposite in that sense to supervised learning. And this machine learning type of problem involves using a model to describe or extract relationships or similarities or groupings in a data set. And some of the examples are dimensionality reduction. So for example, principal component analysis, which you might have come across, or different types of clustering algorithms. So those are unsupervised machine learning problems. And then there's reinforcement learning. So these algorithms, they continuously learns from the environment such that it can take actions that would maximize the reward or minimize the risk. And this is, so usually it's referred to as a reinforcement learning agent. And it's used in, applications such as robotic navigation or different game artificial intelligence, kind of adversarial networks and those kinds of things are come under reinforcement learning. So for our our workshop today will only focus on supervised machine learning. Okay, so and supervised learning. So as I defined, let's look at it in a more formal sense. So it's a class of problems that involves using a model to learn a mapping between input examples and the target variable. So it has, the data is labeled, and we'll see a little just in, the next slide, what we mean by, label data and so here the models are fit on the training data, which comprise of inputs and outputs. So the features and the labels in the training data, and then it is used for making predictions on the test set where the features but the labels are unknown, and that's what you want to find out, like what category or what sort of continuous variable corresponds to the set of samples that where we don't know the labels. Okay, so there are two main types of supervised learning, as we saw. So classification, which is where you a learning problem that involves predicting a class label, the categorical variables like cancer versus not cancer. And regression is a supervised learning problem that involves predicting a numerical label. So for example, a BMI or blood pressure and so on. And this is just, so this is, just to show you a general setup of how this works. So we have data that, might have collected previously that has both features, and known outcome. So here the input is X matrix, which is, a sort of comprising of different, P predictors here, and then the outcome is Y. So this is the outcome, and this is our these are features. So this is our altogether. This is our training data, right? And then we have set of features and an unknown outcome here that we want to predict. So we build a machine learning model on this labeled training data to predict the unknown outcome. And this is what is our test data. So that's a little bit of more notational background on what we mean by label data and training and test data. Okay, so so some of the examples of classification problems, right? So there are different, first of all, there are different algorithms to do classification. So there's linear discriminant analysis or random forest analysis or supervised or support vector machines. And some of the examples is like, for example, given tissue gene expression profile, right? Like host transcriptomics profile, and you have to classify the samples based on gene expression profile into different disease groups. So that's so basically you would have some samples on which like whether it's cancer or not cancer, and the other samples that where you don't know which is your test set, and then you sort of predict whether that sample based on the gene expression profiles that it has learned from training data is cancer or not. And then there's regression problems, which is there are different algorithms here. So there's linear regression, which maybe you've come across, and then there's penalized regression. The different lasso ridge and elastic net. These are some of the so regressions where they work on high dimensional data, and that I use these in my research a lot, because I deal with like high dimensional gene expression and microbiome data, and we can maybe when we get to later for the slides, maybe we can dive a little bit into what penalized regressions mean. But some of the examples can be like, for example, predict body mass index from microbiome data, right? So you would have samples with known, sort of microbial abundances and, known body mass index, and then some samples where you do not have the body mass index. So you have to learn that from the training data and predict what could be the BMI of those samples. So that's basically, so that was just to cover, the types of machine learning algorithms and, the supervised learning that we want to focus on. So next let's move on to looking at different steps involved in a machine learning task. But before that, is there any questions? Okay, let's move on then. So this is basically the overall pipeline here, right? That looks at different steps. So first, obviously, we begin with data collection. So, we have the data, then we perform different data processing, feature engineering, feature selection, and then we, after this pre-processing, we have the pre-processed data, the cleaned up data, upon which we do the data splitting, so into train, validation set, and then the test set, and then we use cross-validation to perform training. So we get the trained and, we train and validate data sets, and then we get the learned model as a result of training. And then we use that to predict on the test data here, right, to get, the different types of prediction. So that's the overall overview. And we are going to step into each one of them and see, like, how we do the different steps here.
We have a question.
Okay.
What is feature engineering?
Sure, we are getting there. That's in the next slide. So we'll get into each of these steps, and then we can see how different feature engineering algorithms are there. Is there anything else?
Nothing else right now.
Okay, cool. All right, so so let's first get to the data collection step. So that our data should be suited to answer the learning problem before, and then so you could collect new data. So you have to think about the study design. What is the sample size? Whether you want to have, like, can you get try to have even sampling for case and control samples. And then what is the type of data that you want to collect, whether it's gene expression data, bulk or single cell, microbiome data, or protein sequences, and so on. Or you could also get existing data from repositories. So, for example, these are some genomic databases like dbGaP, which is for genotypes and phenotypes, or TCGA for which is the Cancer Genome Atlas for cancer genomics data, or the Human Microbiome Project, the HMP, to get any human microbiome data on different healthy and disease cohorts. But the point is that this is an important thing to consider, because the quantity and the quality of data is very important for our building our machine learning models, and how what will be the performance of So this is an important thing to keep in mind. And then next is the data processing, right? So how will so this is a critical step, right? How will the raw data be used to build machine learning models? So it's very critical. And then there's a question of like, one may ask how the success of a machine learning algorithm may rely primarily on the data set and not on the algorithm itself. So the explanation is that, the most popular machine learning algorithms are well established, but each data set is unique, right? So each data set has domain specific features. So given the uniqueness of each data set domain, the machine learning can succeed only if we as researchers understand the data set details and we are able to arrange it properly before running any machine learning algorithm. So so that's the kind of so both we have to make sure that our data input into the machine learning algorithm is sound. And so here's different feature engineering steps, right? So or different feature engineering methods, and this is not an exhaustive list. So we'll look at some of these, like one-hot encoding, binning, what is this, and then we'll also look at some of the feature selection. And as I said, these are not like not an exhaustive list, but we are only covering some of the typical ones. So one-hot encoding is one of the feature engineering methods where we transform any categorical features into binary features, right? So let's say, the example is there are 3 types of cancers. We want to look at carcinoma, sarcoma, and melanoma, right? So what if we transform them into numerical encoding, right? So then we'll have carcinoma as 1, sarcoma as 2, and melanoma as 3. And you can see that the specific order here is not important for decision making. Actually, both of these 3 are like, it's not that there is an ordering between the 3. There are 3 numeric 3 categories that are at the same level, right? So transforming into numerical encoding can actually give us incorrect input, because it's kind of hinting to the algorithm that, hey, there is that these 3 cancers are at different there's a gradation, but in this case, at least for this example that here, we want to keep them at the same level. So to do that, we can introduce something which is called one-hot encoding, where you do is sort of you have a binary encoding for each feature, and so if it's a carcinoma sample, you have 1 here and 0 at other places. If it's a sarcoma, if there's 1 here and 0 at other places, and so on. So you can see, so you basically you have your feature vector and then a feature matrix, right? And then you have added on like these 3 sort of new columns, right? Instead of having disease or cancer subtypes as carcinoma, sarcoma, and melanoma, we now have 3 columns. So it result in increasing dimensionality of feature vector a little bit if you're trying to sort of have this as part of your model. However, it does not imply any incorrect ordering and thus avoids any misleading prediction. So this is one of the one of the ways of sort of one of the tricks of feature engineering. And next is binning. So this is basically just, you want to summarize features sometimes where you don't care about the exact value. So for example, you could maybe convert a numerical feature into categorical feature. So for example, age, which is a numerical feature, you can convert it into age range categories. So for example, 0 to 18 years is, age bin 1, 18 to 35 years is age bin 2, and so on. So basically what you did is like replace the age feature with age bin categories. And in some, and again, you don't have to do it all the time, right? Like, but this is when you sort of need categories if you don't care about exact age year that you want to predict. So it depends on your prediction problem, but this is in some cases also done. Another example of binning is could be to transform microbiome features, which is like microbial sequences, operational taxonomic units, or the OTUs, into genus level, right? So this is basically this evolutionary relationship between the microbes, which is represented by different taxonomic levels. So it starts with kingdom, phylum, class, order, family, genus, and species. And then OTU is like a little bit, kind of more granular. Sometimes it's not to get into too many details, but so there's different taxonomic relationship or different taxonomic bins in which microbial sequence can fall into. So you can perform that taxonomic binning and, sort of only consider microbial sequences at genus level or at family level. So we basically take the values at the species level, for example, and then you sort of bin them into like order level or something like that. And we'll see some example of doing that when we do our hands-on tutorial. And the idea here is, why would you want to do this? So this can reduce your dimensionality, right? And it can also reduce a little bit of redundancy, and you can sort of, so you could build your models at the OTU level, but many times I've found that, like, collapsing to a higher bin or collapsing to a different category gives it a better prediction performance. So yeah, those are some examples. And so this overall, the goal is to hint to the learning algorithm that if like a feature is falling within a certain range, the exact value does not matter. Okay, so those are some, okay, yeah, and then there is normalization and scaling. By the way, any questions so far in these features?
We do have a couple questions. The first one is from Anup Ambikan. He asks, how do you decide or calculate the optimum sample size?
So that's a very good question, actually. So I guess there is, when you do study design, you can do some power calculations. And I guess there is, I think there's a way in R you can do that too. So yeah, so that you perform power calculations. So like, what type, what values of effect size you want to see, and that kind of thing. And based on that, you and given what type of data you have, what type of questions you want to ask, and what type of effect size you want to see in your analysis, so you work your way backwards to understand that. And then there are other considerations, right? Like, if you have different disease groups, you want to make them as even as possible, because sometimes uneven, at least in terms of machine learning, say classification, like uneven sample sizes could be a problem, especially if the class that you want to predict is like kind of is low, has low representation. So
and we have one more question from Priya, who asks, could you please comment on the normalization of data from two different organisms, such as a human and a mouse, before putting them into the model, or data acquired from two separate experiments or from two different platforms? Do you use a specific method, or do you have any suggestions?
That's a great question, because there is, so I think if you have data coming from two different organisms, I'm not sure. I mean, so first of all, the concept of biologic, so there are two things, right? Like if it's coming from different platforms and different sequencing technology and all of that, right? Even if it's the same type of data, right? Like if it's, let's say, human fecal microbiome data, right? And it's coming from different platforms and different sequencing and different, all that. So the technical effects is resulting in results in the batch effect, right? You might have heard of this term, and I don't know, like there are ways you can maybe control. Sometimes I've seen like SVM techniques that talk about, like, at least for the microbiome data, to remove batch effects. So there are some like domain specific ideas, but really it's really sometimes the batch effect influence can be so much that you cannot actually get rid of the the technical, the technical noise there. So yeah, so I mean, so that's the problem of batch effect when you have like, coming from different platforms. So you can check, like, in your field, if you're working microbiome or gene expression or whatever, if there are batch effect correction techniques, or and one of the ways is, like, I sometimes just add them as covariates in my model, right? So to control for them. And yeah, so those are some thoughts there. About mixing data set from different organisms, I'm not sure, because now this is like a, biologically very different signals, and I'm not sure whether you would want to, and depends on what type of model you're building. But for example, like if you build a model on like human gene expression data, right? And then, you want to, or rather on mice gene expression data, and you want to predict something in humans, I don't know, maybe it could work, but I don't think that would be a good idea to, combine the feature matrices from different organisms, because again, we have to think about the biology, unless, you're the way you have designed your experiment, you can like have, sort of overlaps, or you can see a, sort of some commonality. So, as I noted before, like, start from the biology, like, does this make sense that, whether you would build model on one organism and predict on the other, or something like that, right? Or maybe it's a different question you have in mind. I'm not sure what exact research problem you're looking into, but think about the biology a little bit. And otherwise, just using some statistical approaches to normalize, I'm not sure whether it is going to, sort of give you, that I'm worried it might give you some misleading results. So again, we can chat more, and maybe you can describe the question a little bit more in like email or something. And but yeah, those are my sort of first thoughts on that problem. Cool. Anything else, or should we move on?
That's all the questions for right now.
Okay, cool. Thanks, Sabrina. All right, so yeah, and then the next feature engineering type is normalization or scaling. So we want to, sometimes want to convert the actual range of values into standard range, so between -1 or 1, or between 0 and 1, depending on what kind of so you'd have to do the data exploration to see what range you want to have. So and again, biologically, does it make sense? Like I cannot, yeah. So right. So there are inputs. So this ensures that the inputs are roughly in the same range before machine learning algorithm is applied, right? And then there is so one of the ways to do this is min and max scaling. So you basically take away, as you can see in the formula here, right? So you take away, you find the minimum value and in the feature matrix, and you subtract that from each features, and then you divide it by the difference between the max and the min values. Yeah, so that would be your kind of the min max scaled data. And then there's different transformations for skewed data. Like if there's data is following some log distribution, you could use some square root transforms or logarithmic transforms. And so this is quite domain dependent, and you have to look at the distribution of data and yeah, it's for your type of data set, like, you have to look at, what are different normalizations that people use in the field. So for in microbiome data, there's specific ones that like, sometimes use logarithmic and square root combinations. So, and then there's standardization. So this one refers to scaling feature values so that they obey properties of a standard normal distribution. So where you basically take the mean to be 0 and the standard deviation to be 1. So the question is, whether you want to standardize or normalize, and so the most supervised models, they benefit from standardization, and standardization can help with any data set with outliers. And yeah, sometimes just you can try different ways and see which one performs better or actually makes more biological and sort of technical sense. Yeah, so those are some feature engineering ideas there. And then we can move on to feature selection. So basically the idea is you want to identify any informative features, that is, remove any irrelevant or redundant features or noisy features. And this can lead to better learning performance. So it can have higher learning accuracy and low computational cost and better interpretability of the model, right? And there are different methods here. So filter-based methods, you specify some metric to filter out any noisy features. And this, in most cases, you have to do this. If you're dealing with high throughput data sets. And then there's some wrapper-based methods where you consider selection of features as a search problem. And then there are embedded methods where algorithms have inbuilt feature selection method. And we will quickly take a look at some of these things. So filter-based feature selection. So this is very domain dependent, and there's prevalence and abundance-based filtering. So you basically filter out genes with expression values less than X, for example, in fewer than Y samples. So this is, just to give you an idea of what kind of filtering you can do to, keep relevant features. There's, forward and backward selection. I'm not going to get into details of this. You can look it up, and maybe we can sort of also provide some additional information after the workshop on if you're interested. And then there's embedded feature selections, as I mentioned, like some machine learning algorithms have an inbuilt regularization or feature selection methods like lasso, ridge, and so on. Yeah, so again, I'm seeing we are running a little behind on time, so I'm not going to jump into these details of these features. So next is data splitting.
I'm sorry, we have 3 questions, and
cool.
They shoot here. I'm trying to get through. The first one is, does min-max scaling improve the algorithm's efficiency, or does it serve another purpose?
I think so it's again, this is you're just transforming the data to sometimes it can have an effect on the efficiency and performance of the machine learning. So yeah, you have to be careful, just so look around it in your data set and is it difference, for example, difference in gene expression, or if this is actual abundance or actual expression value. So yeah, yeah, you have to pick the method appropriately and see whether it helps the performance. So most of the times, just putting in raw data, untransformed data is not suitable for most machine learning algorithms.
The second question is standardization or normalization the same as scaling?
Yeah, I think didn't we? So I think the normalization and scaling are interchangeable, but and sometimes transformation is also used for this, but standardization is again, this is just a terminology thing. Yeah. So
the last question is, does normalization happen to the entire data set or to each feature individually?
Yeah. So basically, you apply it to your entire data set, right? So every feature gets scaled or transformed, whatever. So yeah, so it's applied on the entire feature matrix. So each feature gets transformed like that. Yeah. So you obviously don't do one transformation on certain subset of features and another one. You want to keep the same transformation most of the times on everything. Yeah. Cool.
Sorry, we got one last question in. Hendrik just wants to confirm the main purpose of doing feature engineering is for dimensional reduction, or is there any other purpose?
Dimension reduction, and then also distribution of your data, right? So you want to make sure that dimension reduction is, definitely one of the things when you do feature selection, right? But or sort of binning type of thing. But with standardization and normalization, we are also looking at like different sort of you're making either the data normal or like so, or you're scaling it by something. So it's both. Cool. All right. So let's move on. So next step is, we want to look at data splitting. So we generally don't fit the model on the entire data set, right? Because we want to build a model. We don't want the model to do well at just predicting labels of the samples the learning algorithm has already seen, right? So what we really want is an algorithm that's good at predicting on the samples it has never seen before. So ideally, we split the data into 3 sets: the training set, which is used to fit the model, and it's usually the largest fraction of the samples. And then there's a validation set, where you evaluate the model fit on training data, and it's used for hyperparameter tuning. And then there is the test data. So you want to, check the unbiased evaluation of a final model fit on the training set. And there are different split percentages. People use this, 70, you could do a 70% training, 15% validation, or 15% test, but you can play around with this. It depends on the number of samples and what are the number of hyperparameters you need to tune and so on. And yeah, so usually but most of the times we don't have enough samples to do this 3 splits. So what we resort to is cross validation. So so we, with lesser data, we just split the training data into we split the data into training and test, and then we split the training data into train and validate sets to simulate validation. And this is kind of what you see here is cross validation. So basically you split the training data into a certain number of folds. So let's say you split it into 5 parts, right, or 5 folds, and then you kind of fix a value for tuning hyperparameter, and then you fit the model on the training set, and then you test on the validation set, and then you get some performance metric for that hyperparameter, right? And then you repeat this k times, such that each split of the training data has been used for validation at some point, as you can see in this sort of schematic here. And then what you do is you average the metric across those k iterations, and that gives you like the cross validation score. And there are different ways to do cross validation. So we have k-fold cross validation, where you have, which I was just talking about. So there's k statistical representative of the broader data set, and your k can be 10 or 5, or so it's called 10-fold or 5-fold cross validation. These are typical numbers. And then another popular one is leave one out cross validation. So this approach will leave one data point out of the data set, right, training data. And if there are, so for example, if there are n data points in the original sample, then n-1 samples are used to train the model, and then the one left out sample is used for validating, right? So every single sample here, as you can see in the schematic, is used as a validation at one point during the cross validation approach. And as I mentioned, so we want to do the hyperparameter tuning during cross validation. So again, what is the hyperparameter? It's the it is what determines the learning of the model. So for example, how many clusters in a k-means clustering, how many trees in a a random forest model, and those kinds of things. So there are different methods, and when we usually, you don't need to implement your own cross-validation methods, but just, so they're linear and grid search. So there could be a list of search parameters, different penalties or kernels, and you could, sort of, for every penalty parameter, you do that cross-validation thing that we just saw, right? And then you identify the performance. Then you do it for another parameter, and then you go on and so on and so forth. But this can be time-consuming and computationally intensive. So there are some efficient approaches to do this, and so random search or gradient-based search, gradient descent algorithm, for example, is a good example there. And then evolutionary optimization and so on. So usually we don't have to implement this, and it's inbuilt in like, a library of the machine learning package in either R or Python or whatever language you're using. So that's that takes care of it. So just to summarize here, once your optimal value for hyperparameter is found, you fit the model on the entire training set with these best hyperparameters, these optimal hyperparameters, and then you assess the model using the test set. So then you evaluate the accuracy and whatever different performance metrics, which we'll see in a little bit. So basically, this is just a very brief overview of like the different steps in the process of machine learning. Any questions before we move on?
Yeah, we've got 2 questions. The first is, in your experience, what models tend to work best for microbiome abundance data?
Yeah. So we'll get to that. So it depends again, right, on what question you want to ask, right? If so, for example, random forest model, which we are also going to apply on microbiome data, works well with sort of classification and also for regression. So you could use it for both. So that's one of the methods that works well. And again, in my research, I use some penalized regression models where basically which does like feature selection, where I'm trying, I'm trying to identify, what human genes are associated with what microbial features, right? What microbes. So there, given the high dimensionality of the data and other statistical issues, the lasso and ElasticNet and those kinds of penalization models also work well. Yeah, so I mean, it depends, what kind of question you're asking, classification, regression, you want to look at like what type of, prediction you want to make. So yeah, those are just some thoughts there.
The second question is from Priya, who asks, how do you decide the learning rate?
Learning rate, which is like is that for a specific algorithm, or like, is that a hyperparameter for, I mean, if it's a hyperparameter, you do cross-validation to identify the different a learning rate, if that's like
yeah, Priya said the hyperparameter.
Yeah, so I mean, that's a typical way I would do it, like just use cross-validation. Yeah, and there are different ways, and tenfold or the k-fold cross-validation, or leave one out depending on how many samples you have, and then there's also stratified cross-validation. They're nuanced approaches, but basic idea is you sort of do this kind of sort of leave out certain samples, and you validate your models on that. Yeah. Okay, cool. All right, so we covered this. Okay, so we covered the process of machine learning. So now let's look at some of the ways we can evaluate the performance of a machine learning task. And here, given our limited time and the type of problems you want to look at, so we want to do evaluation for classification problems, right? So, and one of the typical ways to look at that is confusion matrix, which is a table summarizing how successful the classification model is at prediction, right? So this is like showing confusion matrix for a binary classification task where you want to identify. So this is the actual, whether this is a disease sample or it's not a disease sample, and what was the prediction, whether it was a disease or no disease. And so these are different cells here, so this is the true positive. So these are cases in which our machine learning predicted, yes, they have the disease, and they actually do have the disease, right? So those are true positives. The false positives are when the prediction is yes, but they don't actually have the disease. And this is also known as the type 1 error. And then there's false negatives where we predict no, but they actually do have the disease. And this is also known as the type 2 error. And then finally the true negatives where we where the machine learning predicts no, and in fact the sample is not a disease sample. So those are the so that's the confusion matrix. And then based on those parameters there, we have different performance metrics. So we have accuracy, which is the measure of fraction of times the model predicts the correctly both true positive and true negatives out of the total number of predictions. So basically the sum of all the four numbers here, four metrics here. And then the sensitivity or the recall or the true positive rate, these are, all the synonyms here, basically identifies the fraction of times that the model predicts positive cases correctly from the total number of actual positive cases, right? So, and then there is specificity or the true negative rate. So basically, which is the fraction of times the classifier predicts negative cases correctly from the total number of actual negative cases. And then there's precision, which is the fraction of times the model predicts positive cases correctly from the total number of cases that it predicted as positive. So basically the true positive out of the true positive and false positive. So yeah, so those are, I mean, like it's hard to keep all of that in, mind. So, feel free to, when you're doing this, like look it up online and, sort of, sometimes I tend to forget the, the correct formulas. And I guess, I have a sheet where, all this formula is summarized in one place. So, and then there's receiver operating characteristic, right? So these are curves that plot the true positive rate against the false positive rate at different threshold settings. And we'll see just in a little bit. And this idea is originated from radar engineering and now, is used predominantly in for different like machine learning for assessing machine learning performance for classification. So basically what this shows us is the true positive rate versus false positive rate, and here's the formula for true positive rate, which is also sensitivity or recall, and the false positive rate is basically 1 minus the specificity. So that's that. So basically what the idea is, so let's say we have the test result, right? So the blue are the points with the disease and the red are points without the disease. And then so let's say this is a threshold, right? And where x-axis, so x-axis is predicted probabilities and y-axis is, this one is a count of observations and samples. And so at this threshold, let's say we call these patients as positive and, and the remaining, this side of, are the patients as negative. And so these, hence, this area here is showing the true positives, right? And then this area here, right, which was predicted as positive, but actually is, without the disease, so it's negative. These are the false positives. And then this, which was actually, disease and we identified as negative, is true negatives. And then these are basically, they were disease cases with disease samples, and we identified as non-disease samples. So these are false negatives. So basically, as you move this threshold towards, positive or the negative side, you can get these different values. You can get a different confusion matrix, right, corresponding to each of this threshold. And then whether you move it in the positive or the negative side, you can see the different value, the changing values of the true positive and false positive rates. So basically an ROC curve is just doing this, right? Like it's moving the threshold at different points here. And sort of it's built, so the ROC curve here is built from confusion matrix, which results at different thresholds. And so as you can see, this is the false positive rate and the true positive rate. And it, basically at each point, it's calculating, these values, and then that's where how the plot is generated. So some of the ROC curve extremes are like, the best test example where the distributions don't overlap at all, right? So this is, usually no real world example, you'll find this kind of performances. And the worst case situation is where, distributions overlap completely, right? Like that red and the blue line. So you can't really identify the cases from the controls. And then the associated concept is area under the raw curve. So AUC is, which is the most common, one of the common measures of prediction accuracy for binary classification, and it ranges from 0.5, which is no better than chance, to 1, which is perfect classification. And this can give an overall measurement of test performance. And you can sort of compare different tests, different algorithms also based on difference in the estimated AUC values. So here are some AUC examples, right? So this is when you have 100% area under the curve, which usually never happens, and then you have the area, I mean, usually doesn't happen for a real world example. And then this is the AUC at 50. So this is like just like a random, classifier, which, is like, cannot discriminate at all between the two classes. And this is an, classifier with AUC of 90%. So it's pretty, competent in doing the classification. And this is, kind of an average classifier at AUC of 65%. Yeah, so those are some of the ways we can, sort of determine performance of our classification task. And so before we move to part 2 here, is there any questions?
Yeah, there's a question from Sana who asks, if my accuracy is really high, like 99%, and they indicate that this is for a logistic regression, 10x single cell expression, she asks, am I most likely overfitting, and what can I do to avoid that?
Are you overfitting? So that, I think, well, like if on your samples, one way to look at that would be to, like, I don't know if you're looking at all your samples or like just subset of samples. Maybe if you have access to, so overfitting happens when your algorithm is sort of fitting the model too closely to your sample points, right? So maybe if there is it's possible you can try if there is a data set of similar type on like a separate cohort, but like let's say you're looking at cancer, and if there's single cell expression data on similar type of cohort in similar disease and so on, and try to see. But I mean, maybe the condition is so drastic that you get perfect classification, right? I mean, so I've never heard in real cases to get 99% accuracy, but again, it could be possible. So I would try, like, sort of maybe even subsetting the data sets and trying to see, like, if it or trying a different data to evaluate. So it depends, like how you have split the data, what is your training test size and all of that, right? So yeah, those are some very basic thoughts there.
Next question is, can I use the pre-processing steps you mentioned before doing PCA in unsupervised machine learning?
So for PCA, again, the focus of this, today we are not talking about unsupervised learning, but in general, what I do is, like, for PCA, I will at least remove some noisy features, right? So for microbiome data, just getting rid of things which are like singletons, right? For example, a microbe that only appears in a single sample, right? So that kind of quality control I would still do, but not like, I mean, I generally, yeah. So yeah, you can do some pre-processing there, and it depends on from data to data, right? So yeah, like I think in microbiome data we generally just do it on like just a basic pre-processing before we fit the I mean, we do the dimensionality reduction like PCA or actually it's called PICO-A, but yeah.
Last question is, in the beginning you differentiate the parameter with hyperparameter. Is there any example on setting the parameter, or could you provide an example?
So basically the parameter is set by a machine learning algorithm, right? Yeah, machine learning algorithm learns what. So for example, if I was running, penalized regression model, right? So the penalty term is my hyperparameter, right? Like, what is my, how sparsely do I have to identify the features, right? So that's something that you learn through cross validation that you set before running the machine learning model, right? So, and then once you run the model, right? So then you get, let's say, imagine like, a linear regression type situation where you get like different coefficients for the different features. Let's say I'm trying to identify whether, what microbes are associated with, a gene expression, right? So then, so my hyperparameter is the penalty, like the penalization that I want to, and the parameters are actually the coefficients for each microbe. If to say like, okay, what microbes are actually more or less contributive towards predicting a variable of interest. So anything else? Sabrina, just making sure.
Yeah, that was all the questions.
Okay, cool. All right, so I see that we are over 1 hour mark, and I will quickly go through the the theory part of this microbiome section. I wanted to do just a quick intro to the microbiome, and then get to the hands-on tutorial, and maybe so let's finish the theory part here, and then maybe we can take a 5-minute break and then come back for the hands-on tutorial. So yeah, so let's just to give you a background on the human microbiome. So it's comprises of many of you might have heard of this, but so human microbiome is comprising of trillions of microbial cells that live on and inside us. So there's tens to hundreds of trillions of microbial cells. There are 10 times more cells than human cells, right? And there are almost 3.3 million genes just in the human gut microbiome, which is almost 100 times more genes than our human genome. And there are over tens of thousands of species of microbes on the human body. And so sometimes the human microbiome is referred to as our second genome. And so for today's workshop, we'll focus on human gut microbiome. And so the human gut harbors a complex community of over trillions of microbial cells, which is called the gut microbiome. And these gut microbes include diverse communities of bacteria, viruses, fungi, yeasts, and parasites. Right. And the gut microbiome provides a wide range of beneficial properties to its host, such as breaking down complex carbohydrates, synthesizing vitamins, host immune regulation, and so on. And now, this complex community of gut microbes play a critical role in maintaining human host health. And any perturbations in the gut microbiome have been associated with several human diseases, as you can see here. So obesity, type 2 diabetes, colorectal cancer, and inflammatory bowel disease. So just to give a brief overview of what I work on. So I've developed machine learning models to identify host microbiome associations in different diseases. So here, this work, I built a machine learning method to identify association between gut microbiome and ethnic health disparities in the Western population. And then, and since gut microbiome has also been shown to associate with host gene expression and in some experimental models, and that so host gene regulation and gut microbiome are both associated with disease. So I have also built integrative frameworks to identify associations between host gene expression and gut microbiome. So here I looked at host gene expression microbiome interactions in patients with cystic fibrosis to understand why they might be susceptible to early onset of colon cancer. And then I've also developed machine learning frameworks to identify patterns of interactions between gut microbiome and host gene expression in patients with colorectal cancer, inflammatory bowel disease, and irritable bowel syndrome. And so, as you can see here, again, I don't have time to get into details, but we have identified some shared patterns. So they are shared gut microbes, but they are interacting with disease-specific host gene expression. Yeah, so that's so basically, what I'm trying to say is gut microbiome can modulate different diseases. and because it, sort of interacts with our immune system and host cells, and can modulate the gene expression as well. Okay, so for our tutorial today, we'll only focus on the gut microbiome data. So let's quickly look at the process of obtaining microbiome data. So we get a microbiome sample, it's a fecal sample here, and then we have two major sequencing technologies. So one is the 16S approach, and then so here the 16S approach, what you do is it it targets a specific gene present which is present in all bacteria, the 16S RNA gene, but it can be used to identify different bacteria. So here, again, I'm not going to get into different details here, but the idea is basically you amplify and sequence this gene, and you do the grouping and match against databases to identify the composition, what organisms, OTUs are basically, sort of which microbial sequences are present and at what abundance. And then there is shotgun approach, which basically, you grind up all the DNA in the microbial community in the sample, and you align and assemble the reads and match to databases. And then finally, the for our purposes today, what we get is, as a result, like, the first data that, you have here is the OTU table, which is the operational taxonomic unit, it stands for OTU. And then so basically, this is a table of different OTUs, or the microbials, different microbes representing different microbes here in the rows. And then the columns are the different samples. And each cell is showing how many sequences for that particular microbe can be found in a given sample, right? And so, yeah, so this is an OTU table that we'll also be working with today. And the data set that we're looking at today is fecal microbiome data from 16S RNA sequencing. And it's on a data set with samples from patients with enteric diseases, and there's healthy samples as well. And yeah, we'll be looking at OTU table, and this is just the reference from which the paper from which we are using the data set. Yeah, so and so the idea is like excuse me, we want to classify the enteric disease samples of enteric disease from healthy. Just a second. Excuse me. So yeah, so that's our goal. And to do that, we plan to use the random forest classifier. Excuse me. So basically, so random forest classifiers are built using decision trees. So there, so here's an example tree which shows whether the person's risk of having a poor outcome after they have a heart attack, right? So at each node, there's a yes and no question, which is based on a variable or a predictor, and then you split the training data accordingly. And each node, it asks, like, what feature will allow me to split the observation in the hand that the resulting groups are as different from each other as possible. So so that's like, what a decision tree is. And so random forest is basically, sort of consists of a large number of individual decision trees that operate as an ensemble. So it's an ensemble learning algorithm, and it uses a bagging or a bootstrap aggregation, where it creates bootstrap training sets and fits a separate decision tree to each bootstrap set. And by bootstrap set, we mean random sample of size n, and that's with replacement, and then you get a majority vote from B decision trees that you built using those training sets. So basically, just pictorially, this is what is going on. You have a sample, and then basically every decision tree kind of decides. Here you can see the entries, and then it decides whether it's a classification problem, so it decides whether it belongs to class X or class Y, and then there is a majority voting from each of the tree to finally get what is the prediction of the final class. So that's how that's just an overview of that. And so next, let's take a few minutes of break, like maybe 5 minutes, and then we come back and sort of try to implement the machine learning pipeline on microbiome data. So Yeah, I'm happy to take any questions, but in the meantime, if there's any questions or but so it's what time is it? Around 2:16, right? So how about we meet at like about 2:20? Sorry, I'm in Central Time, so I'm telling my local time, so at 2:21. Yeah. Any questions? I'm happy to answer.
There's one question. What is the difference between bootstrapping and cross-validation?
That's a good question. So Random Forest has its own way of doing this cross-validation approach, in a sense that it does, takes out some random samples and it builds the decision trees based on that. And then it sort of predicts on the left out samples, which is called the out of bag samples. And then, it gives an out of bag error. So you can, typically, you can just do that and also report out of bag error, but like, cross validation approach is also can be applied on top of that to basically sort of make sure, like, this is just like kind of because cross validation is a little bit more accepted way, but I've heard that many people don't even, you may not need it. So one difference I can see is like, as in Bootstrap, is that because you are basically for every tree that you build, you are taking a random set of samples and sort of, and then you build a tree on that training set and then predict on the remain out of bag samples. It may happen that some of the samples kind of overlap between the trees, right? So that's one of the things. And I think, like, sort of boosting trees somehow get rid of that. So, I guess I don't have an answer on which is maybe more efficient. I'll have to think a little bit more on the caveats. But yeah, like Random Forest has its own like internal method of like, kind of doing this out of sample tests validation.
We do have some more questions, but maybe we can wait so you can take a little break before.
Yeah, no worries. And then I also see there are some chats and stuff. Is there? I have not been monitoring that. So if
I've been looking at the chat box that's been coming to all the panelists as well as the Q&A. So maybe you got some chats that were just for you.
Yeah, I think we got overall for the general panelists, but if you have some specific, it can be a good time to check.
Right. Yeah, I don't see, I guess these were the questions. So one thing that I'm seeing here is your accuracy could look really high if you could have an unbalanced. Yeah. That's also true. Yeah. That's a very good point. So Helena Van Tol mentioned in the chat that yes. So there was one question, right? Like, what if you're sort of. So maybe we'll wait for 2 more minutes. As we are still talking, and so that why your accuracy can look very high is because you could have an unbalanced set. So if you have like everything, so the algorithm can try to predict everything as the the positive, the majority class, right? And then you think that, oh, your performance is so good, and that's why there's it's called the accuracy fallacy. So just don't look at only the accuracy numbers. You should also look at the sensitivity of the model, the specificity, all of that, right? And then see whether, maybe the model is very poorly sensitive, but it can just easily just predict everything as majority class and get by, right? So that's a very good point. Thanks, Helena, for pointing that out.
Tatiana, for the next section,
Oh,
just in case that there
hold
are
on.
some.
Oh, sorry. So there will be a hands-on tutorial after this, and then after that is Tatiana's session. Yes. So maybe go ahead if you
I was
want.
just going to say, Sambhawa, for your next section where it's more of
Uh-huh.
a workshop, and then there are
Yeah.
some questions that are more specific to the code that I might not, I don't know, there might be some questions that are just maybe not best asked live, but maybe responded to that are about errors and things. I was just wondering, Tatiana, would you be willing to kind of chime in on some of those?
Yes, I will monitor that, so no worries.
Okay.
Okay, so yeah, and like, you know, if there's so I'm going to sort of share my screen here, and as I change to sort of the console. So I hope everyone got like the so you guys can see my screen, right? And I hope you got to download the data, and you know, where is my website? I can try to pull out the so I hope that all of you were able to look at the GitHub page. So I'm trying to pull this out right now. Let me see. So basically all our data is all the data and material for the session is here. And so right now what we're using is the session one's you know, data. So there is data in the input folder, and then there is the code here, and then here is all the information. So I hope you got it. And so I hope you have it. But so just make sure to put the two data sets here in the input folder, and I guess that's it, right? Like that's the structure. So we can get started here if everyone is ready. And you know, if you have not been able to download, don't worry, we'll share, you know, all the code and stuff. So it should be okay. Is there any question before we begin? Any quick questions?
There is a question that's asking what the input data looks like, but I think
Yeah,
once
sure.
this person downloads the data and sees it, that'll become clear.
Yeah. So right, so we'll actually just take a look here in the console. So can you guys see the screen well? Like, I mean, is it visible? Should I expand, or, you know, just let me know?
Yeah, maybe would you mind making it full screen?
Okay, is this good?
Yeah.
Better?
Yeah.
Okay, sounds good. Okay, so basically I can't I'll have to change the thing a little bit. Okay. Right. So as I said, this you know, and you know, this is the data taken from Singh et al. Paper from 2015, and you know, it has microbiome data from patients with enteric disease, and as well as healthy control. So let's, you know, start. So I hope you have these libraries installed. If not, you know, you can use install packages. So we're going to load these libraries here, and you know, I'm using RStudio, but doesn't matter. Like, you know, you can also, so this is just to make sure, you know, our we set the different paths using relative to the current directory. Okay, so first is getting the metadata here. So that is the file, and so you know, basically this should give us different understanding of the different samples. And you know, we can also take a quick look at so you can so basically what this is showing is these are different sample IDs, the status, whether it's a patient or healthy. There were different pathogens. We are not necessarily looking at this, but yeah, they also annotated that the sample has, you know, salmonella infection or Shigella infection, so on, and the different time points. And then this is just basically saying that the disease state is enteric, diarrheal disease, or healthy. And okay, so let's let's do some dissect, some data exploration here. So what is the, so as we saw, there are, so this seems like there's more samples here than or rather like the disease and healthy samples. And so there are also 2 time points at which the data was collected. And basically, because in that study, they also did a follow-up of some patients, and that's why there might be 2 time points. So we just want to, for our purposes today, we just want to use the first time point. So we do subset by the time point, and then we only are left with those many samples there. So basically now we have 201 disease samples and 82 healthy samples. And this is our classification problem here, to identify EDD versus healthy. And then so let's input now the actual data here, which is the OTU, the microbiome data. So this is the OTU table. And then we check the dimension just to, so these are the number of OTUs here, and these are total number of samples. So obviously, we have to, sort of subset to identify common samples between the metadata and the OTU table. So we first identify the common samples, and then so those are the 282 samples. And then we want to make sure that like our samples are in correct order. And then what is the dimensionality, and that all our sample, I mean, this is just to make sure all samples are lined up correctly. And then, yeah, and so next we want to, get to the pre-processing step, where basically, we want to filter out any noisy features, right? So one of the things is like, for example, you could have different things here, but one is to remove any OTUs with which have fewer than 10 reads, because those OTUs might be noisy. And so we do it here, and what happens is, as a result, we are left with only 1,500 or so OTUs. And then we sort of look at, also we want to remove any OTUs which were present in fewer than 1% of the samples. So this is basically also another, sort of quality control sort of, we want to make sure that the OTUs which are also, sort of prevalent in enough number of samples. So we do that here, that filtering. And so then we are left with about 1,300 OTUs. And next, what we want to do is, sort of we want to bin the OTUs to the genus level. And we'll see how we go about that. So basically, the different microbial sequences, right? Different OTUs might be coming from different genre, and what we want to do is we want to take those microbial sequences from the same genre and then collapse them at the, at that level, at the genus level. And we do that by summing their abundance counts. And so first we want to, sort of identify the different taxonomic levels. So we do that with this step here. And we append the OTU table. Oh, so one of the things that somebody had asked was to look at the original data. I mean, I've already filtered this, but hopefully, it doesn't matter so much. So we can basically take a look at the OTU table now with the, so these are our different OTUs here, and here I'm only showing, so basically this is showing you like the label at different taxonomic levels. This is like a Parabacteroides OTU, and this is like a this is belonging to genus Doria, and so on. So here what I did is like I split this label into the 8 taxonomic levels we have, and just added genus level. And these are the, as we had seen in the schematic in the slides, these are our different samples. And these are the different abundance values. So like, you have the sort of the Patabacteroides, this thing is abundant. This is the abundance in this particular sample. And as you can see, many, so this is like a very characteristic of microbiome data, which is pretty sparse. So it's like there's zeros. So what this means is like very few microbes are actually shared across different individuals from which these samples are coming from. So that's pretty typical. And Bacteroides is, one of the prevalent ones. So we kind of see it found in every sample. So yeah, that's just, sort of eyeballing the data, which is also a very good idea. Okay, so basically what we have done here, another thing I want to point out is, so we have like, split this thing, the labels, and like just showing the genus level. And then, so what you can see is that, like, for example, for Bacteroides, right, like there's, so there are multiple bacterial sequences here with the belonging to genus Bacteroides, right? So Bacteroides here, and so on. Or like, or this taxa here, Lacnospiraceae, so we see this multiple times. Or Pseudomonas. I don't know if there's Pseudomonas right in the same frame, but, so the idea I'm trying to say here is that, we want to collapse it at genus level. So for that, what we're going to do is we're going to sum up any abundances that belong to a specific genus level, right? So we are going to like kind of sum up, for example, for Bacteroides, all the Bacteroides occurrences, and so that will give us the genus level abundance. So that's the an example of our binning or summarization. So we do that. So yeah, we also want to obviously remove any OTUs where genus level was not characterized, so that we are left with 944 OTUs at that point. And then we take the, so here is where we are doing that summarization, so we are trying to aggregate the abundances at the genus level. And then this is just attaching the OTU name. And so, and then that's the OTU table that, sort of our genus level table. It's no more an OTU table. So if we look at this right now, basically it gives us for different genre, like what is the abundance, right? So we saw, for example, Bacteroides, so this is now having the sum of all the Bacteroides microbes and so on. So that's the one that, so we wanted to, sometimes the reason is like, I'm not showing that here, but if you can try, you have the data. So you can try building the model at the OTU level and then try comparing it with the genus level model that we have here. So, and you'll see that there's a difference in performance. So maybe, more noisy features appear. And so at the OTU level, so by summing them up together at binning them at the genus level helps improve our performance. And so yeah, so here is like, one of the transformations is to basically calculate the relative abundance. Basically what you want to do is like basically take the abundance of every genus, and then you divide by the value of the total reads for that genus in every sample. So basically what we are saying is you take basically every, so in a sample, right, these are the different bacteria, right? So we want to, convert into relative abundance, meaning like taking abundance of each bacteria and then dividing it by the sum of all the microbes here in that sample. So you're basically, yeah, so you're showing the relative abundance of every microbe. And so we calculate that here, and now we prepare the data for training. So the Caret package likes the data not in the, currently it's probably in the data frame form. So we convert it to data matrix. And then also, most machine learning algorithms like rows as samples and features as columns. And here our feature is the genus, the microbes. So we will also do that transformation. And this was, this was just to make sure that, like the healthy is the control, like, the reference level. Yeah, and so that's the that's the data. So now that's ready. And now we have to do the next step, which is the training and the test. But before that, any questions so far?
There aren't any questions in the box right now.
Okay, cool. All right, so let's yeah, so I hope you're also able to do this along, and if not, you'll have the code. So the next step is we want to do the training and the test. So first, we want to set the seed so it's like reproducible. So first want to set the split the data, and here we are using 80% training and 20% test. You can, and this is just a little bit of explanation on what everything means, and you can actually go look up this function in the Caret package, and you can find some more things you can do to set this train index. And this is in case you want to create 10 different 10 different subsamples with 80% training and 20% set, and then you want to average it out, the performance. So but here we'll just do it for one 80-20 split, and then see. So here I just wanted to see, like, after we did this, we randomly took 80% of the samples, right, and put it in the training data, and remaining. So this is just to show, like, in what the different so this resample, like, is showing the different there are some data points which are in the training, but like they're also and the remain, like, for example, some samples are in the test. So this is just to show that how that random sampling works. And yeah, and so then we take the strain index, and then we do the we subset it to get our training data, and this is basically to get the label of our training data. And then and then similarly we leave out those train indexes to get our test data, and then what is the labels for that. And now we want to perform the cross validation. So that's the train control function there, and we're using the cross validation method, and we're using 5-fold cross validation. You can also try 10, or there's another setting to set it as LOOCV for the leave and out cross validation. And this is just to make sure there's we can compute the area of the ROC curve later on, and this is to just make sure that we keeps the scores of the model using the ROC. So that, like, train control again, like, has like a giant list of parameters that you can set here to tune how you want to set up your training, and you can tweak that, and but here we'll just keep it simple and straightforward right now. And yeah, so we set that, and then we set the seed again. This is just for reproducibility, that we can all have the same result. So now we're going to do the training step. So we give it the training data, and this is the so these are the training features. This is the labels of the training data. We're using the random forest, and instead of default metric, which is the accuracy, we are setting it to ROC. You can, again, this is just an example, so you can play around with different metrics here. And this is just to say, like, this, we are using the train control that we have used for the cross validate 5-fold cross validation. So we can perform the training now, and what we get is the basically sort of different. So it takes a little bit of time, hopefully, so yeah, basically it's just showing that, like, so it's trying to mtry, which is basically the number of variables that are randomly sampled as candidates at each split. So random forest also, instead of like taking all your features, it also has like a randomness in feature selection that it does for sort of keeping the decision trees a little bit uncoupled. So anyway, so that's that variable, and it's trying to split at different levels, and this is the different ROC the ROC values, the sensitivity, specificity, and so on. And this, I mean, this was just to show that like across our 5 folds, what was the ROC and the different metrics here. And then basically what we get is as a result at the most, like kind of the average of the across the 5 folds. And then so our model is trained. So now we want to use that to do the prediction on our test set. So so we do this, and then what we find is, this is basically showing not very meaningful right now, as we can see, like, it's just showing the labels of the, the samples which were, what is the predicted, whether it's healthy or disease. And so we can build a confusion matrix, right? And that will be a little bit better to see. So, okay, so this is our actual, this is the 39 EDD sample, and these are the predictions of between EDD and healthy. And so, we find that like they're different. So, again, you can go compare the confusion matrix labels, where is the true positive and false positive and so on. But, you don't have to compute them by hand. So we can compute so we can use this to get all the different metrics. So, this is our sensitivity, the specificity, positive predictive value, the many, and then the precision, recall, and so on. So you can, and then the balanced accuracy that Helena had mentioned in the chat before you. So there are different these are, so you get all the different metrics here, and yeah, so just look at every, so it depends on what you want your classifier to be, like, more sensitive or more specific, and so on and so forth. Like, it depends on like what question you're asking, what kind of data you have, and so on. And if time remains, we can talk about nuances of how to deal with like unbalanced data. So we yeah, so and then this is just to sort of get compute the probability instead of like the predicted class value. So basically what it gives us is like the different basically just giving like the different samples have different, what is the probability of predicting it as EDD versus healthy, and so on. So it makes the call accordingly. So this we are doing so we can compute the ROC curve. So basically we get the AOC as the area under the curve as almost 96%. And we can also sort of plot this. So you can go ahead and execute this, and it will create a plot right in the session 1 folder if you have something like that. If you created the folder like sort of we talked about, so we can go I'm trying to pull out the plot here, the ROC curve, and that's over here. Sorry, so I'm trying to pull it over to my screen, and so that's what it looks. Oh, come on. It won't let me share this. Oh, because it's in full screen mode. So anyways, you can see the snapshot here, right? And we can also, I can pull out that plot. Let me try to minimize this. And so yeah, so this is our ROC curve that we get, and you can try to play around with like different different cross validation folds, and try to tune different parameters and see if like and try to plot them, and you can compare the different methods. Even you can try a different algorithm if you wish, like gradient boosting, or any method of your choice. So that there's a lot of this is just just to demonstrate a working just a working example. And then what we want to identify is like what are the features, what are the microbes that were important in, doing this, the classification. And so for that, we compute variable importance. And so because we're using caret here, so, in this package, how it is computed is that basically the prediction accuracy on the out of bag proportion of the data is recorded. And then the same is done after permuting the predictor variable. So you compare the prediction accuracy on the true data, obviously like the out of bag proportion, the ones that were not used in the building the training model. And then you do the same after, permuting the variable. So you basically, sort of just break the relationship in the data and try to see whether, what is the accuracy then. And then the difference between the prediction accuracy on the true data and the permuted data is then, is averaged over all the trees, and then it's like normalized by the standard error, which gives us the variable importance. So that's what like the varimp is doing. Basically what it tells us is like, this is a way to say on the true data, this was the accuracy, and when we permuted it, this is the accuracy. And what is the, the bigger the difference, right, the more important the variable is, like the in helping predict the, in helping the machine learning algorithm. And then, there are different ways, there's different, impurity metrics or Gini index and so on, that we are not covering here today. But this is one way to see, like, what variables might be driving our classification. So basically, so we get the important variables, and this is just to capture the importance column, and then we want to sort them by the decreasing performance, and then we get the top 10, so that we can look at what are our top 10 features here. So as you can see, like the so we get Clostridium cluster 4, I think is one of the pretty predominant bacteria in the gut. And the Clostridium in general, like, comprised of both like beneficial and disease associated bacteria. But this one probably is beneficial. So maybe it is significantly reduced, right? Might be significantly depleted in the disease versus the healthy samples. L-steeps is microbe that is mostly associated with, has been associated with number of disease samples, and also seen in like enteric diarrheal diseases. And then so some of these kind of make sense, and like so this might be like maybe overrepresented in the disease samples versus healthy. So we can, obviously we can do more data exploration and like, sort of plot these values and see, what is, if it makes sense, or, and then Blauschia here, which is also like a beneficial microbe, like butyrate producer, and could be sort of maybe significantly reduced in disease samples, and so on. So yeah, so this is just, to give you an overview of the entire the entire pipeline that we talked about of implementing a machine learning model. Yeah, and so you're free to sort of change, like tweak the models here, and and have fun with at least this, this one data is just an example. So yeah, like I think that's most of it on my end. Is there any, I'm really happy to take any questions at this point.
There's a question that says, hi, do you have any books or reading to suggest to learn more, especially considering data types and adjust parameters in addition to attempts?
In addition to what?
In addition to attempts.
Okay. Yeah, sure. I mean, like, so I don't have it right now, but what we are going to do is we are going to sort of on our GitHub page, we can add additional resources. So we can suggest some books, and obviously, like, if you go through the slides, there are different books and papers that I'm citing. So those could also be very useful, especially like Introduction to Statistical Learning is one of the books I often turn to. And then there's also some advanced books on data science and machine learning that could be beneficial. So yeah, I'll be happy to post that after and I'll also share the slides after both of us, Tatiana and I, will share the slides after. So should have some resources.
A couple more questions. Do you control for background noise in the data set, such as removing biological signal that could come from a DNA extraction kit, like, for example, contamination?
That's a good question, right? So this one is fecal sample, right? So most of the times there's very less contamination issues, but that's a great question which comes up in my research, where I look at colonic mucosal biopsy samples, and there's the chance of high, because it's low biomass, right? So fecal sample is very high biomass. So low biomass samples tend to have contamination, and there are different ways to, control that. And we actually did that in the cystic fibrosis project that I briefly mentioned. So one of the ways is to look at, make sure, the lab reagents are not in your samples, is to use negative control. So you can use both positive and negative control. So basically you can have a blank, and again, this has to be done way before you build your models, right? When you're doing study design, and that you have some blank sample sequence, meaning which is just water or just the reagents. And then you sort of sequence that, and then you you compare the composition of the true samples with the negative and the positive control. And there's methods to so decontam is a great package to do that, at least for the microbiome data, which can sort of help you identify if there are any things in your negative controls which are or in your blank controls which are overrepresented, and you have to sort of get rid of those. And then there is then you can also do it a little bit more manual way, which also we sometimes have to come down to, because so there's also list of some well-known lab reagents and such contaminants. So basically, yeah, you can also filter that. So in this data set, usually with fecal sample, it's not a problem, but yeah, it happens in tissue samples. So yeah, so those are some approaches.
So we have about 5 more questions, but I was just wondering, in the interest of time, since Tatiana's session, I think, is supposed to start soon.
Yeah.
Sambhawa, maybe you'd prefer to answer those by just typing the answers in the Q&A.
Yeah.
Would that work better for you?
I think, yeah, that's a great idea. I can just type the answer when during Tatiana's session. Yeah.
Okay.
So maybe we can take a 5-6 minutes break before Tatiana can start, right?
Yes, sounds good. So yeah, I suggest we have a 5-minute break and see you all at 3.
Yeah, sounds great. Thanks so much, everyone.
Stay tuned.
Stay tuned. Yes.
So this is the second session that is devoted to the viruses. A little bit. Okay. Yeah, it's working. So a little bit about my journey. I'm a mathematician from a strong Russian mathematical school. As a student, we were taught, we were trained in computer science, and we had access to supercomputers, so we can apply fundamental mathematical methods to different areas like medicine, biology, economics, and social sciences. And many of my fellow students, they were very interested in information technology and computer security. But I took one course in bioinformatics as an undergrad, and it became a turning point for me. After that, I went to industry and worked on several projects related to software tool development and we analyzed heterogeneous data. And then I decided to go back to academia and get my doctoral degree in bioinformatics and computational biology. And I was very fortunate to meet Professor Bowley, who is a very strong mathematician, who encouraged my interest to apply mathematical method to biomedical challenges. And we were particularly interested in recognition system in biology, like CRISPR-Cas mechanism, that is adaptive immunity of bacteria, and also host-parasite interaction, in particular between viruses and bacteria. And we apply set theory and linear algebra method, in particular matrix method, and I will talk about it a little bit. And also artificial intelligence and machine learning methods to various biomedical challenges. My part of the workshop will consist of 3 parts. First, I will give you a brief overview of viruses in microbiome and their diversity. Then in part 2, I want to discuss with you 3 research findings that I think exemplify the complexity of relationship between different microorganisms, and in particular, they show the role of viruses in this relationship. These 3 examples are when bacterial viruses can trick human immunity. Also, viruses can restore healthy guts. I think many of you heard that bacteria can do that, but viruses seems also have this capacity. And viruses, and also I'll talk about the viruses and cancer microbiome. And in part 3, we dive into the exploration of virus-host interaction. I'll give you a little bit of theory in terms of mathematical methods and how they can be applied to exploration of virus-host interaction. And then we try this on our own in the hands-on parts on the workshop. Okay, so viruses. Usually when people heard about the microbiome, they think about bacteria. They can be beneficial or can be pathogenic, but microbiome is much more than that. And Sambhawa mentioned it, except for bacteria, they can be fungi, they can be archaea, which is also microorganism, prokaryotic microorganism, but they are very distinct from bacteria, and also protists. And each category of this organism has their own viruses. And in microbiome, we have a mixture of human viruses, animal viruses, plant viruses, fungal viruses, bacterial and other viruses. And all of them can alter the state of this microbiome. So make sure when you talk about microbiome, you keep all this hidden complexity in mind. And now, as I mentioned, I was really interested to share these 3 findings that changed my perception of the role of viruses. And these are great examples of the complexity of these viral host interactions. I'll first with the first example, when bacteria viruses that cannot infect human directly can actually trick human immunity and help bacteria infect humans and animals. In this case, we will talk about the Pseudomonas aeruginosa. It's a gram-negative rod-shaped bacterium, and it can be present in microbiome without causing any symptoms for the healthy people. But if patients are immunocompromised, it can cause problems, for example, cancer patient and cystic fibrosis patient. Also, these bacteria often colonize burns and chronic wounds. And the problem with this bacteria is that many clinical isolates are often antibiotic resistant. And unfortunately, there is no approved vaccine available against this pathogen. Also, some strain of this bacteria, they have a prophage. Prophage is a virus that is inserted in the bacterial genome. And under some condition, this prophage can leave the bacterial genome and form a separate particle. And oftentimes when this happens, it will activate the lytic cycle when virus replicate and kills the bacteria. However, some phages can escape bacterial cell without causing this damage to the bacterium. And this filamentous phage is one of them. And previous research demonstrate that often this phage is found as a structural element of biofilms. Also, previous findings demonstrate that it can play a role in inflammation in lung infection. In this particular case, we are interested to see what role this phage plays in the chronic wound infection. The data include 111 human ones, and out of those ones, 37 was also colonized by Pseudomonas aeruginosa, and 25 of those were also contained the prophage in them, and the rest 12 was prophage free. And as you can see on the left, this is two ones, one infected with Pseudomonas that doesn't have a prophage, and it's A, and B is Pseudomonas with a prophage. And you can see that it's pretty distinct. And the one infected with a bacteria without a prophage looks much healthier. And if you look at the how the infection progressed, that infection caused by Pseudomonas aeruginosa with a prophage resembled much more cases when these ones are not healed in 6 months. So in this case, they much severe outcomes and they cause much more problems for patients. To test the role, to explore the role of this prophage in the infection, so we et al. They used the murine model, the mouse model, and so they wound the mice and then they allow for 24 hours to heal, and then they inoculate it with bacteria that also has a luminescent label. So after 3 days, they check, and if the one was colonized by bacteria, they saw this luminescence. In this case, they infected wounded mice with 3 different pathogens. First, it was this Pseudomonas aeruginosa without a prophage, so the prophage was deleted. Then the prophage was deleted, the bacteria with deleted prophage in the genome, but with introduced of this phage like in the environment. And also the original strain of bacteria that has this prophage in them. And you can see that there is a pretty distinct difference with and without prophage. And it doesn't matter, is it in the genome or it's in the environment. And also wounded with infected with bacteria that has a prophage in them resulted in much higher rate of infection. And also it was associated with much higher rate of morbidity and mortality and different the weight loss problems in mice. So it seems that this phage definitely plays a significant role in the progression of this infection. And I don't dig into neither detail of how they demonstrate it, but they come up with a model. In this case, if bacteria didn't have a prophage, yes, and of course I have a reference, so feel free to read more about this. I think it's really outstanding research. So here, in this case, if bacteria didn't have a prophage, it's trigger antibacterial immunity and the infection was cleared. But if the prophage was introduced, then it triggers antiviral response in human or animal cell, and this antiviral response also can suppress the antibacterial response. And in this case, the bacteria can sneak into the ones unnoticed to the human immunity. They also explore this prophage code protein with its potential to get a vaccine against this prophage. And it did work. In this case, vaccination against this code protein of the prophage also was protective against the wound infection, which is great because in this case, you can get a vaccination against this prophage and it can improve the outcomes for patients with chronic wound infection. So this research demonstrates the direct pathogenic role of phage, the bacterial viruses in bacterial infection in animal and humans. And they also show that vaccination against phage might prevent bacterial infection. And it has broad utility for chronic wound infection and cystic fibrosis and for cancer patients. Interestingly, that there is other filamentous phages for different gram-negative bacteria like Salmonella enterica and Escherichia coli. And in this case, many of them are also antibiotic resistant. Another important application from this study is that phages, even though they can be used for phage therapy, when you can use prophages to kill bacteria, in some cases, bacteria can have these phages on a leash, and you need to be really careful when you pick the phages for this therapeutic application, especially for antibiotic-resistant bacteria. Okay, case 2. In this second study, it's explore relationship between enteric eukaryotic viruses and intestinal health. So it's not now it's a animal virus, but it's has an unexpected role in human health, human intestinal health. So this particular study performed by Kernberg et al. In 2014, it studied the germ-free mice. It's mice that doesn't have any microbiome in them, so the intestine is sterile. And they introduced the virus, the murine norovirus, to these mice, and they show that it doesn't lead to uncontrolled viral replication and disease in them. However, when these mice develop, without any bacteria in them, they have a very many problems with their immunity and the development of the intestine. And this researcher demonstrated that murine norovirus can compensate for these observed abnormalities in germ-free mice. It can even reverse, introduction of this virus can reverse abnormalities. And also it can lead to increase in granules and lysome positive cells, and also in a number of immune cells like CD4, CD8, and T cells. However, this impact was specific to mice that had depleted bacteria, because in the conventional mice that have normal microbiome, they didn't have this pronounced effect. However, in antibiotic treated mice, it do help to induce this, to reduce these intestinal abnormalities due to depleted microbiome. And this effect of the norovirus was similar to this effect that bacteria have separately. These findings suggest that virus can restore intestinal morphology and also lymphocyte function without any adverse effect like inflammation or disease. And it can help animals with depleted microbiome due to antibiotics. So it can be beneficial in restoring or compensating for these deleterious effects. And also it's interesting that viruses has capacity in terms of shaping intestinal immunity like commensal bacteria. And the third example that I want to discuss with you is viruses and cancer microbiome. As I said, this microbiome is very complex, and it has human-specific viruses that many previous studies show that they are strongly associated with cancer. And it has other eukaryote viruses like animal viruses and plant viruses, and also the bacteria phages. And all this together impact the microbiome significantly. So in the next study, they look at the colorectal cancer, and which is really a devastating disease. According to National Cancer Institute, in 2016, over 1.5 million of Americans were diagnosed with colorectal cancer, and over 500,000 died from this disease. And the question is, can we come up with an accurate and non-invasive method for detection of this colorectal cancer based on stool samples, taking into account bacterial and viral community? Maybe it can help to facilitate early detection and prevention of colorectal cancer. And also, it's interesting to understand the role of gut viruses in this disease. So this particular study was done by Hennigan et al. In 2018, and they had data from 90 human subjects that has 3 stages. Some of them are healthy, then have adenoma, which is a benign tumor that is not a cancer, and also carcinoma, that is cancer. And they analyzed both bacteria and viral data. For the bacteria, they used 16S RNA gene and shotgun sequences, and they used viral purification. And they analyzed only materials that were enclosed in the capsid, viral capsid, to reduce the possible noise from the DNA that's floating in the environment. And then they use random forest classification to separate between those 3 states. And you can see on the left, there are just bacteria, which is denoted in gray, the performance of this model. Red indicate bacteria together with viruses, and orange is viruses alone. And if they take into account the whole metagenome data, in blue, there was a lot of noise, and it was not indistinguishable from just random guessing. So the take home from this slide is that definitely bacteria and virus has some information about this disease. And if we combine them, it significantly improves the performance. Also, when they analyze different viruses, majority of them were lysogenic. So it means that they can incorporate themselves into bacteria. And they also observe strong association between bacterial OTUs and viral OTUs, which speak about the relationship between close relationship between viruses and bacteria that are present in microbiome. They also built networks of this interaction based on abundance and importance, and they suggest the following conceptual model. So when bacteria and virus both introduce into intestine, there is some consensus that should be achieved in this microbiome, because viruses eliminate some bacteria and it alter the bacterial composition. And then they start to form this closely related community. And then some of the bacteria can also penetrate the intestinal wall and have some interaction with the host cell, like human cells. And that's what Sambhawa explore in her microbiome research, like how these bacteria affect the expression of genes in the humans, how it's interfere with that. However, that's also has a response, and it's also form a community. And if the particular phage introduced, they can perturb this community, and they can increase or reduce the disease in patients. So it seems like viruses definitely present some component that should to be taken into account when you analyze this complex interaction between that can result in health and disease. So in this case, there was strong association between virus community and colorectal cancer, and viral community was primarily composed from the temperate bacteriophages. And also they found that there is phages that what they call influential that can alter the bacterial community because they can affect multiple bacteria at once. And it can be more promising approach for altering bacterial community than just directly modulating the influential bacteria, like the bacteria that like Pseudomonas aeruginosa or some pathogenic E. coli. If you target them precisely, it has much less broader effect than if you just do it through the phages. Okay, I hope that by now I demonstrated you the complexity of this interaction and show you that the virus definitely should be taken into account when you analyze microbiome data. And so based on my research, what I'm doing, we develop computational method to explore this virus-host interaction. And it's interesting. In this case, the data that we analyze is complete genomes, and it's usually look like text. And when you analyze genome, you often so it looks like this. But what researchers can do, they try to break it into parts and then organize those parts by, for example, color and size. But when you look on these parts alone, it's really hard to infer the initial complexity of the genome present. So that's why you need to be very cautious when you, in your analysis, focusing on the particular gene, like separate pieces, because you might be missing a lot of information how those pieces fit together to organize this complex picture. So my research is focused on the recognition system and biology. And this system, like CRISPR-Cas system, work based on string recognition. And they recognize exact string in a genome. And if here on the left, you see in gray is the space of all possible strings that exist. And bacteria, depicted by dictionary of bacteria, occupy the subset of this all set of strings, and so does the virus. And the question is if these two subsets overlap. And you can use set theory and matrix method to analyze the interaction between these sets. And one of the main questions that we're interested to understand, and how does this intersection change if you change the string that you recognize? Because many biological systems, they have very strict range of the string that they recognize. For example, CRISPR-Cas system, they recognize strings of length 20 to 40 base pairs. A little bit of math. I don't want to scare you, but it's just annotation that I will use throughout the presentation. So here G is a genome, and cardinality of G is genome size. M, we denote a string length. Basically, imagine that you screen genome with a window of this length, and you store all the strings that present in this genome of this particular length. And U of M is a set of all possible strings that you can potentially have. And because we have just 4-letter alphabet, A, C, G, and T in a DNA form, the cardinality of this universal set or complete dictionary will be 4 to the mth power. And DM of G is a set of string that is present in a given genome. And you can do this for different string lengths, basically from 1 to up to the size of the genome. And the question is, what are the relationship between these genome dictionaries and as genome size and universal dictionaries M varies? Here I give you the example. It's a particular virus, PX 174, and particular strain of bacteria, E. coli K12. And this on x-axis, you see the string length, and on y-axis is a number of unique strings that you observe in a genome if you screen it with this particular window. And this semi-logarithmic scale, that's why this exponential growth look like a straight line. So the gray is the size of this complete dictionary, and the blue is the size of the bacterial dictionary, and red is the size of the virus dictionary, and this is the overlap. And you can see that at some point, this overlap disappear. In this case, M equals 17. It's the longest common string that these two genomes share. And interesting that this genome dictionary has two limiting factor. One limiting factor is the number of all possible strings that you can have for a given length. And it's particularly important for very short strings, because basically for M equals 4, you have just 4 to the 4th power different strings. And no matter how big your genome is, there is not much string that you can create out of this limited, under these limitations. But as the string lengths grow, you can see that the all possible strings grows exponentially, and you have a lot of diversity in them. And these different genomes can occupy different subset of this universal set. And you can separate the two if you have a length big enough to reduce the noise and increase the signal. And previously, there was many important findings based on k-mers, because they often, these short strings, they called k-mers, they often use for sequencing. And based on that, if you go back here, there's short strings, you can separate different genome based on the frequency of these strings. And that's exactly what was previously done. And there was many different similarity measures that can be computed based on frequencies. And also for longer strings, for example, M31, there is a database called Kraken that allows you to have taxonomy identification based on these long strings. However, there is a gaps in our knowledge. For example, why certain strings work better than others. And majority of those findings based on k-mers do not specify why. They are linked to the data and they are empirical, and you cannot directly transfer it to another data set. But are there any general rules that can be applicable? And what factors affect the choice of strings in biological system? So in this particular case, when we analyze genome, the many existing methods for k-mer counting, they have limitation because they are optimized for do the counting really quickly, and you cannot change the data structure or add additional parameter. That's why we come up with them. And however, the algorithm itself for constructing the for doing the counting or constructing this genome dictionary is pretty straightforward here on the left, and it's very fast because the computational complexity is linear with respect to the size of the genome. And if you compute the intersection, it's also very fast. It depends on the size of the small dictionary. So in this case, we decided to use the hash tables that are very flexible, and it's efficiently implemented in Python. And during the hands-on session, I'll show you how this operates. And interestingly, when we run the code on the supercomputer, the problem was, the bottleneck was when you download and when you upload the genomes, because it takes very long. So we decided to use the synthetic genomes. Like you just write a short script and you can generate whatever number of genome, whatever length you want with a given number of nucleotides. So we call it synthetic genome. And we run this method on synthetic genome, and we observe really interesting phenomenon. So here, the example of bacteria and virus pairs. On the left, these bacteria virus pairs demonstrate very similar intersection, like if we analyze their synthetic genome. So it means that basically they are not distinguishable from just noise, because majority of these overlaps just based on the fact that genomes are composed of the same nucleotides. But on the right, you see the example of bacteria and virus that are pretty distinct from synthetic, from the genome that you expect from the behavior that you expect from synthetic genome. Here, this is what you expected using just random genome of the same length and GC composition. And you can see this long tail that you do not observe for the randomly generated genome. And this tail is the signal, because it indicates that the probability of just having this coincidence at random is zero. Is negligible, and this is definitely a sign of some biological interaction. And the p-value is pretty significant, so it's 1/10,000. Okay, and do we have any questions so far? I know we covered a lot.
There aren't any new questions in the chat right now, but
okay, perfect. So in this case, the question is how we can model this overlaps, and can we understand, because here, if you look at these two plots, you can see that there is certain lengths after which you don't observe any spurious overlap. And you can get it from this simulation, or you can come up with a way to compute this threshold that eliminates this spurious overlaps. And all the intersection that you observe above this threshold will definitely be biologically relevant. So that's the example that I show you at the very beginning. It's the E. coli and the phage. And if you plot the probability of observing empty overlap between the genome, you can see that for small string lengths, there is always something in the overlap. But if you increase the screening window length, this spurious overlap faded away, and the probability of observing nothing is very close to 1. And here on the right, I show you the tabular data for this plot. And you can see that for m equals 23, the probability of observing no spurious overlap is 0.999, which is very close to 1. And it's just the one genome. But what if you want to see the threshold for other genomes? To do that, we develop an approach that's called statistical modeling. And in this case, we just sample the genome space with different bacterial, with a possible range of bacterial lengths, which is range from 1 megabase pairs up to 16. And for the virus, from 5 up to 500 kilobase pairs. So this range covers the range of sizes for usual set of bacteria and viruses. We also tried different GC content. On the left, you can see the genomes that have the opposite skew. It means that one of the genome is low in GC content and another is high. In the middle is reference. So when all GC, the GC in both genome is equal 50. So it means that all nucleotides are uniformly distributed. And to the right, you have the genome that's skewed in the same direction. So both of them are low in GC content. And you can see that a different combination of lengths. So the length, longer genome and the similar skew shift the threshold to the right. So it means that you need to pick bigger string lengths to avoid this noise in your data. However, this is based on the simulation. But the question is, if there is some regularities, it means that probably there should be a way to compute this theoretically. And so we developed a theoretical formula that match the probability, that use the probability of a single match between two genomes to the probability of observing spurious overlaps for different string lengths. And to understand it, so you can imagine two dice that each of them is represented genome, and the dice has four sides. It's A, C, G, and T, and the probability of a single match will be the probability of observing A on both dice. And because you roll them independently, that's why you multiply the probability. And any match will do, because both A's, both C's, and etc. So that's you sum up all this event. So also that's the probability of a single match, but probability of no match between two strings of length m will be 1 minus p to the m's power. But here we're talking about two genome dictionaries, and each have one of them have one string in it, and another has two strings in it. So for this probability of observing no overlap between these two sets of strings or genomic dictionary is 1 minus p to the m's power raised to the product of the number of strings in those two dictionaries.
Hi, Tatiana.
Yes.
I have one question. Could you give a brief reminder of what the computational problems that you are trying to solve for this phage section?
The computational problem is we observe bacteria and phages, and we want to understand these interactions. And the way we can analyze those, we can see on the shared strings between the two. And later on, we will use these shared fragments to predict the pathogenicity of bacteria based on its interaction with phages. So this, I'll just demonstrate the theoretical part of how we make the computation and why all of these things would work. Because in this case, when we capture signal of interaction in the genomes, we want to make sure that we avoid these spurious overlaps that do not result in any biological meaning. And that's a way of setting a threshold. Because the question is, if we want to screen for overlap between bacteria and viruses, what length should we prefer? And this gives us a theoretical formula for computing this window. I hope it's clear, but yeah, feel free to ask additional questions. And based on this theoretical formula, we can come up with a lower bound.
Oh,
so,
one quick question, one quick note I had is that just requesting the attendees to please, not ask anonymous questions. So if you could please just, reveal your identity if that's okay with you guys. Yeah, so we would, encourage non-anonymous questions. Thanks.
Okay. So in this case, we can compute it theoretically, and based on this formula for the set of bacteria and viruses that we want to analyze, this length of 40 seems like a reasonable choice. And we just computed based on the theoretical formula. The question is how we can, so just, go into the application. When we analyze interaction between viruses and bacteria, we observe that, for example, some bacteria have much, the interaction between bacteria can be very different. So here on the left, you see the lab strain of E. coli, which is benign. And if you compute the overlap between genome of this bacteria and a set of phages, you can see that there is some phage fragments that are inserted in the genome, but they're minor. But to the right, you see the very highly pathogenic strain of E. coli that caused severe intestinal infection in human. And here you can see that one of the prophages is completely inserted in this bacteria. And the question is it possible to predict the functional properties of the host based on this intersection with phages? And the question is, yes, it's possible. Because previously it was found that slight correlation can be found between pathogenic strain and different phages, but it was very, this degree of the correlation varies across different bacterial genera. And the problem is that can be different levels of pathogenicity. In some cases, just introduction of one toxins, like this Shiga toxin phage, can make the bacteria pathogenic. But in some cases, it's a gradual process when prophages are inserted into the bacteria, and then they can gradually disrupt its ability to produce some essential component and gradually move it to pathogenic state. So here we analyze a set of 2,480 phages downloaded from ENA, and in a bit I'll show you how to obtain those genomes, and 101 different E. coli strains. This data is balanced because it has about 57 pathogens and 43% of other strains like commensal, laboratory, and biotechnological. And the labels for these bacterial strains was obtained from the literature. In this case, we regard this bacteria as pathogenic if it's caused severe infections in animals or humans. And otherwise, it was considered not pathogenic. As I mentioned, we use the screening window of length 40 to capture those signs of interaction between bacteria and viruses. But can we validate this window in terms of sensitivity and specificity? And in this case, we take these two strains of E. coli and different phages and count the number of interactions that we observe between pairs of bacteria and phages for different screening window. And for very short screening window, you can see that basically there is a lot of spurious overlaps between this bacteria and phages. But as the screening window increases, you can see that this spurious overlap diminishes and there is a plateau, after which you start to lose sensitivity because you start to lose signals. So it looks like 40 is pretty good trade-off between sensitivity and specificity. On the other hand, we take this window and screen about 5,000 viruses of eukaryote and against these two bacterial strains. And we found only two pairs that have some overlaps, which can be considered a false positive. So it means that the screening window of length 40 has a pretty good balance between sensitivity and specificity. So we took this window and screened genomes of bacteria and viruses for signs of interaction. And for each E. coli strain, we found no less than 30 different phage fragments in them. And among this large set of phages, many of them has no overlap because they can infect other bacteria. In this case, we reduced the set to only 172 phages, which fragments were inserted at least in one E. coli strain. And we created a vector of intersections with these phages for each E. coli strain, kind of a phage fingerprint. And we use it as a feature vector for the classification. And here I also demonstrate the random forest for the classification purpose of equalized strain. And out of back errors was 13.75%, and you will can try classification on their own. And the question, another question that we want to address was, can we come up with a set of reduced set of phages that can be used as an indicator for pathogenicity? In this case, if you have some other strain of E. coli that you don't know if it's pathogenic or not, you can sequence it and see how it interacts with this indicator of phages. And based on that, you can get an estimate of this level of pathogenicity. Okay, so that's this example of this 6 phages, indicator phages, and you can see that definitely this capture the signal of different types of pathogenicity in E. coli strain. Even though there is some false positives, another interesting things that all these phages capture different aspects, and some only taken together they can achieve relatively high performance in distinguishing this bacteria. Okay, so I think, do we have any other questions?
There are no more questions in the chat right now.
Okay, then I think we can take a short break. But before we leave, let me stop sharing. Yeah, so 3:51. So let's take a 10-minute break and move on to the hands-on experience of trying this pathogenicity prediction based on the data. And you should be able to go on the website and click the link and get the data for the second session of the workshop. You need this zip file. You just download it and you unzip it. And it has 3 folders that indicates 3 steps in our data analysis. So step 1, and I will walk you through those steps. I assume that you have Python 3 installed. It can be any environment that you prefer. In my case, it's Spider, which works fine. If you don't have Python or you have Python 2, it's okay. You can just watch and then run code later on your own after you install Python 3. And also, you will need R to the machine learning part. So I hope during this short break, you will have time to download everything and set it up. And we will start at 4. Hi, everyone. So I hope you had a chance to take a break, walk a little bit. Now we are moving to the second part of second session of the microbiome workshop, when we explore the virus-host interaction and how they can be used to predict pathogenicity in bacteria. So by now you should be able to download the code and data necessary for this part.
Before you begin, there was one question right before we left from someone.
Sure.
And they're asking, do you see any effect on multi-drug resistant property of bacteria with respect to your analysis?
That's very complex question, because in this case, multi-drug resistance, that's one of the possible drug resistance in general is one of the possible functional state that can be evaluated based on that methods. And with respect to method, it doesn't necessarily important if it's just one drug resistant or multiple drug resistant. What is more important, how we can predict this resistance based on the interaction with viruses. And this here, I just show you one of the possible application, because you can not only explore drug resistant or pathogenicity, you can also explore other functional state of bacteria. For example, biofilm formation or virulence or any other method. In this case, you just need to obtain the reliable labels. And for pathogenicity, they are binary, like whether bacteria are pathogenic or not. But you also can use, if you can come up with a scale to measure pathogenicity, you can do a regression as well. But I think that's a really interesting phenomena that we can infer some functional properties of the host based on the interaction with viruses. And I think there is biological reasons for them, because in many cases, viruses carry these toxins or antibiotic resistant genes with them, and they can alter the functional properties of the host. So if we have a reliable method to capture those interactions, we can definitely try different prediction models to infer the functional states of host. Thank you for this question.
If you have time, this person had one additional question. They asked, can we correlate the string length theory to evolution of phage inside the host? And if so, how?
That's also, I think it's, yeah. So the, in this case, the length of the fragment, I think, can be related to the timing of this relationship, because for one of this simple way of linking them is that longer fragments can be indicative for more recent interaction, because there is a lot of rearrangement and point mutation happening in the bacteria that can alter these long shared fragments. And I think there is definitely can be a way to link the length of the fragment with timing. Yes, because the smaller the fragment, there are more phage remnants you observe. Because some of the phages, they can be stuck in the bacteria genome and they will be shuffled and rearranged with the bacteria. And in this case, you cannot expect to find really long fragments.
Nice, thank you.
Okay, so let's start the hands-on part. So do you see my screen? Oh no, I think I need to share the entire screen. Okay. So everybody see my screen okay?
Yeah, we see your desktop and the
Perfect.
one
Yeah.
file explorer open.
So when you unzip the archive, there should be 3 folders, and they link to the 3 steps of our analysis. The first step is data acquisition, then will be data preprocessing in Python, and the third step will be machine learning in R. And usually when you work with either biological or medical data, then 90% of your time you spend on the data acquisition and data preprocessing. And usually only 10% actually spent on modeling and tuning the model. In this case, the data that we are going to look at today represent complete genomes. And if you look, the genome in FASTA format basically it looked like text. So the first string in this file, FASTA file, is the title. So what is the accession number? What is the name of this organism? And is it a complete genome or some fraction of it? And the first line is always denoted by this special sign. So you can parse and eliminate it. And the rest is just the genome itself. And bacteria, E. coli has about 5 megabase per genome. That is why I didn't provide you with the entire set of 101 E. coli, because it will be like half a gigabyte. In your folder, you will find just what we can get into it. Okay, so in this case, the data that we are going to use include two sets of genomes, bacteria and viruses. And if you go to the website, here's the link where you can click and see information about this particular bacteria and phages. So here I have this page open, and this is the European Nucleotide Archive, which is one of the places you can go for the genome. And the good thing about this particular data set that it is frozen. So that for now, because they're moved to this more recent interface to obtain genome, and alternatively, you can get genome from NCBI. But for this particular data set, it's balanced because it has almost the same amount of pathogenic and benign E. coli. So here we just scroll down to Escherichia coli. Okay. I think that's the phages. So this is this indicates all the phage genome that they currently have. And as I mentioned, it's about 2,500 different genomes. And here you download this text file that include the accession number. And this is the file that you have in your folder that is called phage list. This file contains all the accession number for these phages. Another file is phage details. This file contains a little bit of details about the phage, like the accession number, date, and name of the phage, which can be useful when you interpret the data later on. And we do the same for the bacteria. So here where you get phages, and for the bacteria, there is a whole set of different bacteria, but we only need the E. coli part. So if you scroll down to Escherichia coli and just take the genome listed here, it's 101 different E. coli strains. And you can see there is a lot of pathogenic one and biotechnological and different lab strains. And here in the folder, you have the list of accession numbers and also the description for the strain. And you know, when you open this file, it's tab delimited, so it's better to open it in Excel. So if you open it in Excel, so here there is a name, the length, the accession number. And when you what you need to download genome is the accession number. And I'll show you how you can download the rest of the bacterial genome yourself. So you go into NCBI. It's called batch address. And it allows you to download not a single genome, but whole batch based on the accession number that you provided. So if you click choose, and then go to data acquisition. And for example, I want all the phage genome that we have here. And you click retrieve. It will generate a list of genome if it matches the accession number to the database. And the good thing about complete genome that they are backed up in different places. So one in NCBI and another is in MBL, and all can be linked using the accession number. So here it says that we have all this genome. And here you click send file, and you have a lot of choices. You can click summary. In this case, you just get this short description of the genome. If you click here, you can choose whether you want the GenBank format when it contains the annotation for this genome. And the difference between GenBank and full GenBank is the full GenBank also has the sequence, the actual sequence itself. For the our analysis, we need FASTA file. So you click FASTA and you click create file. And then you just choose where you want to store them. And it will take some time. I already upload this file with all the phages to this second folder here. So this is the bacterial genome, and I just give you two examples. One of them is benign E. coli K12, and another is this highly pathogenic bacteria. So you can try to create this phage fingerprints for these two bacteria. And I also create a file with a reduced set of phages. I eliminated the phages that doesn't interact with E. coli strains. So this is this multi-FASTA file that I just show you how to download. And I think it's downloaded. So this sequence FASTA is the multi-FASTA file for all these genomes. And you can cut it into individual genome, or you can analyze it, the whole chunk. So I encourage you to download both sets if you have enough space, using this to list of accession number, and the extraction can be found on the GitHub repository. So if you go here, here the brief description of each file that you might need, and here is the link for batch and trace that you click, and you provide the list of accession number, and you got the multi-FASTA file. And this multi-FASTA file, we are going to preprocess them and compute genome dictionaries for lengths of for the strings of length 40 between these two sets of bacteria and viruses. Any question on the data acquisition?
No questions right now.
Okay, perfect. I hope you'll be able to find those genomes yourself and download them. So next step will be preprocess them using the Python. And here in this folder, it's already contained the output after you run the script. But if you're interested to run it yourself, go to the test folder, and this to script that you need. And this is the multi-FASTA file with 172 phage genome and 2 bacterial genomes. So if you open it like me, I open this file in Spider. The first script called core methods ML and B include some additional supplementary procedure that you use for constructing this phage fingerprints. So one of this is multi-FASTA cutter. So this is the function that allows you to create individual files from a large multi-FASTA file, which can be useful when you need to access the individual files for some reason. It's better than just scroll down through entire multi-FASTA format in searching for a particular genome. The next script is counting GC content. And also when you download genome, one of the important things that you need to check that you download it completely. Because when you download the multi-FASTA file, it can be truncated based on, I don't know, memory limitation or poor connection. And it will not tell you that some genome was truncated. That's why I think it's a good practice after downloading this multi-FASTA file that you check the size of each individual genomes. And also I encourage you to check how many unrecognized nucleotides are in this genome. In this case, you will see that some of the genome are sequenced better than the others, and that's one of the indicators. And also it's good to have the GC content for each genome. So here we can start running the script, and then I can walk you through the rest of the procedures. And feel free to ask questions if you have them right away. So in this case, we import two module. One is this core methods, and we use the abbreviation CM for that. And we also use time to time how much time you use for the execution of each script. Here, input files use this multi-FASTA file that we will cut into individual genome, and then we create a list of phage genome. And for the bacteria, here we only have 2 genomes, but feel free to download them all. And if you have them all, you can use this commented code to run it for the entire set of 101 E. coli. So we use lengths of 40 base pairs to screen for intersections, and we save the results. So first, what we want to do is to create what I call the meta dictionary, because you have multiple phages and you want to compute the intersection for each individual phage with the E. coli. And it's computationally ineffective to compute this genome dictionary for each phages on the fly. So what we do, we just generate a list of this genomic dictionary. And since the phage genome are very small, relatively small, it should be able to fit in the memory on a laptop. But if you want to create a meta dictionary for bacterial strains, it can be tricky because you will not have enough memory to store everything. But it can be feasible on a supercomputer. Okay, so let's run it. The first step is extracting individual genome for the multi-FASTA file. And you can see when I run it, this test folder starts to fill up with individual FASTA file for each phage. And so it's done. And now we start reading in and checking the data quality. And for that, after you got the individual genome, you also got a list of accession number that was found in this multi-FASTA file. And another thing that you should get is this info CSV file. This info CSV file just spit out the statistic for each genome. So that's the accession number, the size in base pairs, GC content, and number of unrecognized bases. And you can see that some of genome has none, so it means that all the bases were recognized, but at the same time, some of them we can see equality right here. So some of them have up to 700 unrecognized nucleotides. So it has long stretches of length M. But overall, the quality is okay. So it means that, and also you can match this actual sizes with the info file that I showed you, because when you download genome, there is a metadata related to this, and it should specify how many nucleotides each genome should have. So that's the way to check that you have all the genome not disrupted for your analysis. Okay, let's go back to the script. So it spent 14 seconds for creating the meta dictionary. And because this timing packaging, they usually measure the clock time. So if you have some processes running on your computer, it can take longer, because that's why I ran the same script previously before I open many different processes. And it took like 8 seconds. And now the same can take longer because your computer running several processes and they can compete for resources. Okay, after we compute this meta dictionary, the next step will be to compute the dictionary for individual bacteria, and then compute the intersection. And you can store them in a list, and then you split them into the file, the matrix of intersection. And because we just have 2 genomes, the matrix will be very small. So it's called 42 E. coli pathogen. If you click on it, this is the accession number for the bacterial genome, 1 and 2. And this is the intersection with each individual phage. So the row is the bacteria, and the column is the individual phage. And the cell is the intersection index. Intersection index is the value, has the value between 0 and 1. It indicates what fraction of the viral genome is present in a bacteria. So if it's 0.04, it means about 5% of viral genome for this particular virus are present in this bacteria. And if it's 1, it means that it's completely inserted. So it's kind of a prophage thing. If it's 0, it means that there is nothing in overlap. And the way you compute the intersection ratio is you take the number of strings in the overlap between 2 dictionaries divided by the size of the smaller dictionary. In this case, it's the size of the virus. And it helps you to compare the presence of different viruses in this bacteria. Because viruses usually can have different sizes, but this relative measure allows you to tell, for example, this virus is completely inserted and this virus is only half of this virus is inserted. Okay, and if you run it on the entire 101 set, you got a much longer matrix that will look like this. So it should have 101 rows, considering that the first is header. So yeah, makes sense. And 172 columns. And also there is an extra column in this file that is called pathogen that contains the labels. And if it's 1, it means that the corresponding bacteria is pathogenic. And if it's 0, then it's non-pathogenic. We don't say that it's benign because it can be biotechnological strain or commensal strain or some lab strain. But in this case, we consider it non-pathogenic because it cannot cause infection in animals or humans.
Quick question, Tatiana. So is this pathogen label, pathogenic or non-pathogenic label provided, or you have inferred it from your intersection somehow?
In this case, these labels were obtained from the literature. So
okay,
you didn't have it in the genome.
Okay.
So what we did a literature search, and for each individual bacteria, we check if it's pathogenic or not. And if it's reported in peer review literature as causing infection in animals or humans, yeah, that's an indicator that is pathogenic. And
Thanks.
when we, yes?
No, I said got it. Thank you. Yeah.
Oh, okay.
Yeah.
And there is, so this data set is small, so you can try and test it, but there is larger data set. But the main problem with them is that usually they are not balanced because the number of sequence pathogen is much larger than the number of any other genome, because people are interested in their association with a disease, and they often sequence samples from patients with a disease. So there is a lot of pathogenic strain and not that many benign strain.
Got it. That makes a lot of sense.
So in this case, this data set is unique. So especially for trying different methods, because it's balanced.
That's great.
Okay, and before we move on, I want to just pinpoint some thing in this core method. So there is additional procedure that you can just look at them later. This is for creating just extracting information from the list, getting the text. Because when you analyze genomes, you cannot just concatenate the strings, because if you look at the genome of E. coli, for example, do you see that it's adapted for human reading? And that's why each string is truncated here with the end of line symbol. And if you just read it in without truncating this end of line symbol, you get a lot of chunks instead of solid text. And when you compute the screening window, it will get erroneous result, because many of them will be shrinked. So make sure when you extract it FASTA file, you remove this end of line symbols before you concatenate the text. Okay, and this is the function for computing the dictionary. And in this case, we're computing the dictionary in the way that the string of length 40 is the key, and the number of presence of this string in a genome is the value. But you can set up a dictionary in a different way. For example, you can, as the value, you can use the location of each individual string. And in some application, it's really useful, because if you take strings long enough, they are unique within a genome. So, and if the string appear, you can pinpoint it to the location directly. And you can analyze it, whether it's in a gene or in particular annotated entities. If you're interested to dig deeper, like why this particular fragment is associated with pathogenicity or other properties. Find intersection. Yes, as I mentioned, this just compute the intersection, and the computational complexity is linear with respect to the size of the smaller dictionary. So basically what it does, it's compute which dictionary is smaller, and then go through all string in the smaller dictionary and check if it's present in the larger dictionary. If you do it the other way, it will be not effective. And this procedure just print out the matrix. Okay, any question about the code or the Python code or preprocessing step? Okay, good. So after we compute this feature vector and we obtain the label from the literature, we can move to the next step, and we can try machine learning. So here you have two CSV files. One is the one that I just showed you with 101 different E. coli. And the smaller one, I will talk about it later, but it contains only 6 indicator phages that we detected in the data. Okay, and here RMD file contains the code, and you can open it in RStudio or in plain R and just execute it here. But I already kneaded it in the HTML file, so it will be easier just to walk you through. But feel free to just copy and paste the corresponding fragments of the code into RStudio or R and execute it along the way. And ask question if you have any problems or needs some clarification. So basically what it does, we use the library random forest and caret, and I hope you already had a chance to install it. In this case, you just load them, and then you have this pre-computed file with the feature vectors and labels, and you just read it in, and you can view it and make sure that the dimensions are correct. It should contain 101 E. coli and 174 rows. Why is that? Because the first, you have 172 phages plus labels, and also the first column is the accession number for bacteria. So make sure before you run the any of the machine learning classifier, make sure that you chop this labels first, because it cannot be considered as a feature vector, and it will add noise to your prediction. So that's why we remove this in CBI-ID column first, and then check dimension. And also when the R reads in the file, it's usually take it as a numeric. But for the classification, if you don't do this step, the random forest by default will try to run regression. And it will ask you, are you sure that you want to run a regression because you have just two labels, two classes? To avoid that, you can explicitly say that you want to treat it as a factor. And if you do that, when you run the random forest or other models, it will enforce classification models right away. Okay, so that part resembles the data reading and conversion. And now we want to split it into the training and testing. And here we are not going to play. You can use the validation and play with different parameters, but for simplicity, we just use the training and testing. And for 80% of samples, we put in training and 20% in testing. There is multiple way of doing it. One of them Sambhawa showed you, but here you can just do the sample, and it will pick random sample of the specified size from your data. In this case, it will be 80, and this 80 bacteria will go to training set, and the rest will treat it as a testing. Make sure that you set the seed so your result will be reproducible, because if you don't, each run of the model will produce a slightly different result. Then you link this sample to the data training and data testing. Basically, what was chosen became training, and what is not, what is left over become testing. And then you run the random forest. And here you specify the feature vector and the labels, the vector of labels. So the labels are in our data. That's why we use -173. So we want to get rid of the labels in the feature vector. And that's the label. And we use the default for number of trees. And this MTRI parameter, we set it to 6, and I'll explain later why. But MTRI parameter gives specify how many features you tried on each split. And usually recommended, you try to play with it, and recommended value is square root from the number of features. In this case, we have 172, and the square root will be a little bit over 13. So 12, you can try 12, and you can try half this number, and later I'll show you the results. But for now, we just use 6. And then you call the model, and you can see some characteristic of the model. Here is this out of bag error, and it also provides you a confusion matrix. And Sambhawa explained you how to interpret this confusion matrix. In this case, these results are predicted, and this is the ground truth. And for each class, it's also provide the classification error. It's basically the number of correctly classified entities divided by the total number of entities classified as this class. And the same is true for the second class. So we did the training, and now we can use this model to make a prediction on the test set. And here the parameter, if you use response, it will give you the class label. And 0 is non-pathogenic and 1 for pathogenic. But in this case, the classifier will give you the probability, but you won't get this if you use response. You will just get the class. And it means that it used the default threshold, and usually it's 0.5. So everything that is below 0.5 will be treated as 0 class, class with label 0, and everything that's above will be treated as 1. And we're okay with that. So we save and we can use this code to code the confusion matrix for this. And here you can see the performance on the test set. And you can compute the accuracy, or you can compute other metrics that Sambhawa told you based on this confusion matrix. For now, accuracy is about 85%. You can so you can compute it based on this confusion matrix, or you can also alternatively you can use this code, which give you the same result. Basically, it's the average of cases when your predicted label matches the class label, the actual label. Whichever, so you can use either. Also, there is a built-in function that can do it for you. And depending on the package, so you can try different and choose whichever best suit your needs. As I mentioned, you can get the probabilities because in some cases, when you go back to the confusion matrix, you can see that here the model predicted pretty good, was in predicting non-pathogenic. And there was no any false negatives, but there was some false positives when the model predicted that these bacteria are pathogenic when they actually were not. And depending on your particular task, the cost of different misclassification error can vary. And that's why you maybe need to adjust your threshold to make sure that you take into account different costs for misclassification error. But in this case, we are okay with equal weight for false positive and false negatives. And we want to know the probabilities for each. So we can adjust the threshold if necessary. In this case, you just co-predict, but instead of response, you choose probability. And then you combine your prediction with this, with your label, with the true labels to get this. And if you do just data test pathogen, it will cause some confusion because it's a factor, and when you print it, it will output level of a factor, and it will be 1 and 2. To avoid that, you need to transform it into numeric in this way. And that's why it converts your levels back to this normal value, 0s and 1s. And so this is the probability that this particular bacteria belong to non-pathogenic bacteria, and this is the probability that they belong to pathogenic, and they add up to 1. And this is its true label. So if you scroll down, the probability of being pathogenic is very high, and it's indeed pathogenic. You can use this information to plot the ROC curve and compute different matrix with different choices of this threshold. For example, if you choose a threshold of 0.5, it will give you different labels if you result labels. So that's why it's not a good idea to just always use the default and output only labels. Make sure that you will look at the probabilities and plot the ROC curve to see if the 0.5 threshold will work for you. Any questions?
No questions in the chat right now.
Okay. So next, as I mentioned, this MTRI parameter can is the way to tune your model and see which number of features result in a better split. So here I try different values. Here we tried all possible features, square root that is usually recommended, and I already show you 6 features, which is half of that and half of half. And if you look at the model performance, we use the same split and only different number of different value for MTRI parameter. And you can see that out of back error, if you use all the features, is much smaller. It's like twice as little, which is good. But at the same time, using all features can be computationally ineffective and can overfit the model. But if you use square root, it's higher than what we achieve with 6 features. And if you reduce it to 3, the out of back errors is similar. But in this case, if you run it on different splits, this vary a lot. So 3 feature based on, you can play with it and see how it turned out. But I can tell you that based on the previous running, 3 features is not enough. So this out of back error will float if you use different splits. Yeah, I see we have one question or comment.
Oh, somebody had raised their hand, so I was mentioning. Okay, maybe it was an error. That's okay.
Okay.
Sorry.
So yes, this MTRI parameter help you adjust your model to. Okay, let me know if there is a specific question or if you can solve it. It's okay. And based on that, you can see which performance is how you can compare the performance of the model. So in this case, we choose MTRI 6, like most stable and good in the data. However, you can also try, and here I just show you one split, and you can try with different samples and different splits, but also you can use the cross-validation. And going back to this question that Sambhawa had before, even though the random forest already have built-in bootstrapping, it can be good to use cross-validation in some cases when you want to make sure that every sample will be participated in testing and training. Because random forest, it did bootstrapping with replacement. It means that the same sample, even though it appears just one in the data set, it can be picked multiple times, and it can bias your data in some way. And also, you cannot make sure that all the data set, all the samples in the data set were used for training and testing. But if you use cross-validation, you split your data in such a way on this number of parts, and you make sure that if it's 10 cross-fault validation, it will be 10 parts. And you make sure that each part will be used for testing of the model, and the model will be trained on the rest of the parts. It's, I think, less prone to biases because some splits can be particularly unfortunate or fortunate, and it can bias the performance of your model, the perception of the performance of the model. So make sure that you try different splits and see how your model performance vary. So in this case, we use tenfold, and you use this. There is multiple way of doing it. There is built-in function, but I like this fold function that give you the splits, and you can always go back to each split and see what samples appear in this split and what are special about those, and analyze the splits. And then you use this lapply to run the training and testing on each element of this fold list, and you return the results. So here is the accuracy for each fold, and you can see that it's very a lot. Not a lot, but to some extent. Sometimes it's 1, sometimes it's 0.7, etc. And here you compute just average in 10 folds, and that's what you report. So in this case, it's 0.86, which is okay. And in the same way, you can test different measures and compare the matrix, whichever you like. And another important observation based on experience with working with biological data, sometimes, there is fallacies of accuracy, but sometimes it's the best you can do. Because, for example, with these viral host interactions, we applied a similar method to infer host from the overlapping between bacteria and phages. And the problem with that is that for each virus, you usually just have one reported host, even though one virus can infect many hosts. And when you evaluate the performance of your methods for predicting hosts, you can just measure it against this one host. And the problem is that if it's hit or miss, you cannot interpret the false negative or false positive because you just don't have information about the interaction with this particular virus and other hosts. So the only you can do is that, did you guess this one annotated host or not? And in this case, you just count the number of hits versus all the number of pairs. Because if it's not a hit, you cannot tell that it's false negative, because it can interact, but you just don't have that in a database because you store just one host. Any questions?
Yes, we have a question from Priya, and they say, how do you deal with missing values?
In this case, what do you mean when you say missing values?
So Priya, do you mean like, is it if you have missing features or missing labels, or feel free to type in. We are watching the.
Because in this case, when you construct the feature vector, there is no problem as a missing interaction. So it's just interacts or not. And you computed for the problem of missing values is appears on this data collection steps. And with this procedure of data collection and preprocessing, you don't run into this particular problem. But it can be, very prevalent, especially in medical data set. And there is not much you can do about it. You can just analyze the type of missingness, and is it just particular variable, or you have a different patterns of missingness in your data set, and based on that you can make corrections. Because in statistics there is several levels of missingness. Like it can be complete missing at random, or missing completely at random, or missing not at random. And depending on what type of missing values you have, you need to make corresponding corrections. But I think there is a lot of resources on how to detect this. And that's why before you start analysis, make sure that you look at the data and you understand the completeness and consistency of your data. And that's why I encourage you to always do the data quality check, and including missing values. And if they are, make sure that you could detect the type of this missing value. And then based on the type, you can figure out what result. Sometimes you you have not much choice. You should just remove them. And it works if you have very large data set. it should be okay. But in small data set, removing can be not a good choice because it's left you with very small number of samples. In this case, other way you can try imputation, but you should be very careful with that, because you can introduce the bias in that can result in some erroneous interpretation later on. So yeah, missing values is a separate topic that can be a discussion of the entire workshop. I hope I provide some guidance. But yeah, that's a huge problem, especially in medical data set. Okay, and the last thing that I want to show you is how we can choose the indicator phages. So the question is, okay, we can make some estimate of pathogenicity based on this phages, but what is the contribution of each phage? And can we identify a subset of phages that are most important for making this prediction? And maybe we can ignore the rest. And the good thing about random forest that it can rank features based on their importance. And Sambhawa showed you already this. So what we do here, we use the same seed. And I just want to demonstrate you that if you use the same seed and the same step, you will get exactly the same split and the performance. So make sure that you use this when you're building and analyzing your models. And here there is a function called importance, and M is the name of our model. So if you click importance of M, and I give you just the head, the top six, and they are not arranged. Here you output just a section of this important phages. So they are not. It's not top 6 most important. It's just a glimpse on how this looks like. And here we use mean decrease in Gini. And Sambhawa, she thought about the mean decrease in accuracy. And it's different measure of the importance. Here it deals with the impurity. And if you want to learn more, I encourage you to read about it. And the random forest gives you both. And you can arrange your features based on mean decrease in accuracy or Gini. And here I give you the printout of this different phages. Here is the phages, and here is the importance based on mean decrease in Gini. And this top phages, that's what we're interested in. And you can see it's just for one split. But if you do it multiple split or you do cross-validation, you will get slightly different results. And what we did a 10-fold cross-validation, and we picked the phages that appeared in all of them, like in top 10, I think, top 10. And then we used that phages for prediction. And you have a file that I also supply you, this small file with these 6 phages. It's just some matrix of this larger matrix that contains only these 6 phages, the fingerprint. And when you feed these 6 phages into your model, let me go back to our head into your model and use the same split, you can see that the model performance of just this, sorry. So it gives you a much better performance because some phages contain a lot of noise, and this performance of the model with these 6 indicator phages were much better, a little better than than if you use the entire set.
So, quick question, Tatiana. How did you pick these 6 phages?
we pick them based on their importance.
Got it. Thanks. Sorry,
We compute the importance for each
Yep.
code, and then kind of compute the consensus, like what pages were on top in all of this folds. And it's interesting. I'll show you. So here OOB it's 7.5, and if you go back when we try different MTRI, here if you give all the features to the random forest, it can figure out the 6 important phages.
Yeah, that's great. So basically what you did towards the end is like, just rebuild the model just using those phases, but that's
yes, and
like
just to
not
show you that it's relevant. So
yeah.
You can see this indicator phages, and it's computationally is more effective to just compute. If you need a prediction, you don't want to compute the intersection with all the set of phages. You can just take this 6 as the most important, and it gives you a good estimate of the pathogenicity of this bacterium.
Got it. Thanks.
So that's the idea. It's kind of using small subset of phages that are indicative about the pathogenicity in E. coli strain. And you can use this subset to effectively estimate the pathogenicity in strains. Okay, any question? I think we have a couple more minutes. If you have any other question, feel free to email me. Or yep.
Yeah, we can probably wrap up, right? Like if there's any more questions, if we are happy to sort of, so we will post all the slides, right? After the, so all the code is there, the data is there already on the GitHub. So we are happy to post the slides. And then as there was a suggestion, we can post any additional resources that might be helpful for the reading that will be posted on our website. So please go check back the website and it will be indicated where we have it. And then also, and feel free to reach out to us if you have any.
I'll just, So here are emails.
So we will put it right. Yeah. So our emails are there.
There's our email. So feel free to reach out to us with any question that will pop up after.
Yeah, absolutely. Yeah, please feel free to reach out. Any questions? Yeah.
Thank you very much for your attention.
Yeah.
And good luck with your research endeavors, because I think microbiome is really interesting field, and there is a lot of unknowns. So we hope that you find our workshop helpful in making progress on your research. Good luck.
Yeah. I hope this gives people a beginning point to look at some of these build machine learning models and do fun research. Yeah, thanks everyone.